Circle, circle. There you are. Okay, we are ready. Can you see my screen?
Yes.
Yeah? Okay, excellent.
Yes.
Okay. All right. So let's get started. So today we have a very one of my favorite topics, muscular system and the nervous tissue and brain. Okay. So muscular system, how much we know about muscles? So probably you already know about what in bioscience we was talking about the fascicles, the fibers and all that. And we are going to add more material. We are going to have, as I said, 10 to 15% of the class is going to be a small review. It's not going to be taking long. But you already know the material, but definitely I need to go over. At this moment, I want you to listen to this. All right, listen to this. So we have the electrical impulse that are going to depolarize the terminal axon and is going to release the acetylcholine. The acetylcholine is going to look for the neuromuscular junction for cholinergic receptors that are going to depolarize the cell membrane, releasing and making sodium enter into the cell. The cell then is going to propagate together with the electrical impulse, and they go into the T tubules. In the T tubules, they're going to get into sarcoplasmatic reticulum. In this sarcoplasmatic reticulum will be released the deposit of calcium into the cytoplasm. And these calcium are going to get the troponin, releasing the actin and the myosin to produce muscular contraction. That sounds...
I'm impressed that you said all that in one breath. That was a lot of information.
And one thing I want to tell you, Mr. Mark, I promise that at the end of this part of the lecture, you will be doing the same. That's my challenge, and you will do it. Okay? All right, so are we ready? Are we getting ready? Arby, you get nervous. You like it, right? You like it, right?
Yeah, I like it.
All right, so you will be actually doing the same at the end. You will see. Just follow me step by step. We are going to do a small review, and that is going to, you need to get ready, okay? Put your seat belts, and we are going to start, okay? You're going to get into the next level from today and the next lectures. All right, so let's get started. All right. So we have the muscular system. You already know that we have 600 muscles. That is the pathway we are going to do, chapter 9 and 10. And here we have something that you are familiar with. So what I want for this lecture and for the next lecture too, we are going to use all these words, all these terminology. So you need to be very, very aware, very clear, and put it big, like your first and the last name. All right, so let's start. A smooth muscle is called the involuntary muscle. This is very important. Involuntary muscle, and or smooth muscle. The striate muscle have different names. The skeletal muscle, the voluntary muscle, and the cardiac muscle will be the basically cardiac muscle, right? So cardiac muscle, in addition, is involuntary, obviously, right? The cardiac muscle, listen, your heart, can you stop your heart? You can't. So actually, that's involuntary muscle. And the characteristic of cardiac muscle is this. To make the cell is going to contain characteristics of the smooth muscle. What is the characteristic of the smooth muscle? They have only one nucleus, at the difference of the skeletal. So they have multiple nuclei, the skeletal, but the cardiac muscle have similar as the smooth muscle. In addition to that, the cardiac muscle have similarity now with the striate muscle, because the heart are going to have some striations on the cell. Now, what is the difference between cardiac muscle and the rest? Is that they have here what we call the intercalated disc. Here we have the intercalated disc. This intercalated disc is so important because that is going to have a very strong attachment between cells. And why do we need that? Very simple. They are going to pass the electrical impulse, and that is going to help to the passage of the electrical impulse to make the cells of the cardiac muscle contract, lobbed up, lobbed up, lobbed up, lobbed up, lobbed up, contraction and relaxation, correct? All right, so that is things that you need to remember: involuntary smooth, skeletal, voluntary straight, and the cardiac muscle. But now I'm going to make emphasis in something about the smooth muscle. So we have the smooth muscle are going to be very important for many pathologies that you're going to see later on. One of these that I want you to remember is the smooth muscle are located in the arteries and veins. Arteries and veins. Arteries and veins. If you see here, this is an artery, the artery in a cross section and the vein in the cross section. We have the three layers of the vessel, but in between we have here the tunica media, and the tunica media is composed by what? By a smooth muscle. Why is important to know the smooth muscle in the arteries? This is what is responsible for the vasoconstriction and the vasodilation. Why is important to know about the vasoconstriction, vasodilation? Very simple. The heart, when you have blood passing, for example, in the artery that is this diameter, the blood is flowing, flowing, and flowing. But if you have an artery like this, the blood is going to flow, but with a lot more effort, more difficult. So what is going to do the heart? Is if you have less space to flow the blood because there is vasoconstriction, the heart need to pump harder. The pump need to be pumping harder because the tubing are very narrow, and the same volume need to pass through every second. So that's why. No, I don't think so. No. Okay. Huh? Yes. Okay. So vasoconstriction. The vasoconstriction, what is going to do is to make the heart pump harder. So that means that the blood pressure go up. The blood pressure go up. If you dilate the vessel are going to flow, vasodilate, the vessel are more diameter are going to make easy the blood flow going through the vessel. So actually the heart need to pump less harder. So that is going to basically decrease the blood pressure. And that is what you need to remember, the smooth muscle in the arteries and veins. So I give you just one example that we will do in the next classes. But definitely that I want you to, I want you to focus on that. But this is not the only place that we have a smooth muscle. A smooth muscle we have, for example, in the GI tract. In the GI tract, the walls of the GI tract. The walls of the GI tract, they have this muscle. If we open the stomach, we have muscles here. These muscles are distributed in different way. I'm not going to talk about that, but actually produce contraction. That is, and why we have this contraction in the stomach, just to mention, is that they are going to just mix everything together with the gastric acid to make it a better digestion, correct? And the peristalsis in the small and large intestine, they are going to pass the content towards directing to the anus in order to actually, the peristalsis are going to project forward the content. That's when you have poo, for example, the rectum are going to distend. Why? Because it's going to be full pushing by peristalsis more and more and more fecal material. And when the rectum is distended, that is going to reflex back, contract. Why? And that is as a reflex. And that is going to make you feel that you need to go to the bathroom. The same with the urinary bladder. The urinary bladder is smooth muscle. Urinary bladder is smooth muscle. The urinary bladder collect about 40 drops of water of urine every minute. You're going to see in physiology. And they're going to distend. The capacity of the urinary bladder is about 800. But when they reach 400, that is about the half of a bottle of wine, they are going to produce the reflex of contraction. And who is making the contraction? The smooth muscle. So a smooth muscle, a smooth muscle, a smooth muscle. Another thing that are having a smooth muscle are going to be the ureters. The ureters is the passage of the urine from the kidney to the urinary bladder. They have peristalsis too, because the ureters are going to have actually are going to have a smooth muscle. All right. So, for example, you can dilate or you can constrict that ureter. For example, if what about if I have a stone that is nailed in the ureter? That's a kidney stone. You cannot pee. They can lead into kidney failure if you don't treat it. So what we do is to relax that smooth muscle and make it the diameter bigger in order to make pass the stone. Same in the gallbladder. In the gallbladder itself have a smooth muscle. The ducts that are going to transport the bile towards the duodenum, they have a smooth muscle. The pancreatic duct that are going to drain the pancreatic juice into the duodenum is going to be a smooth muscle. Everywhere is a smooth muscle. And those are the highlights I want you to remember. So smooth muscle are very important. All right, so we okay with that?
Yes.
Excellent. And remember, those are involuntary muscle, involuntary muscle. All right, so here we have what we already know very well. We have one muscle fiber. Don't get confused with the dietary fiber. Dietary fiber is what you eat in your vegetables, your plants, the fiber that you eat in your food, right? That is totally different thing. Fiber we call here the muscle fiber. Fiber is equal to say one muscle cell. One muscle cell are going to be this. The typical actually a striate cell. So this is a fiber, and that is a muscle cell. The muscle cell together, if you have many pencils together, like grab it like this, like 10 pencils together, each pencil is a fiber or muscle cell. All together are going to be called fascicle. We have multiple fascicles all over. And if you put all the fascicles now together, that is going to form a muscle. Now, the endomysium, we have this plastic wrap or membrane that is surrounding each structure. This plastic wrap or membrane that surround each of the cells of the muscle are going to be called the endomysium. So each of them. So each pencil that you have, each one pencil is going to be surrounded by a plastic wrap. Then you put all pencils with plastic wrap together, and this package are going to make all together are going to have a package or plastic wrap again, and that is going to be called the perimysium. And then if you put all the fascicles together and put a plastic wrap around the whole fascicles at the same time, all together like this, they are going to be called the epimysium. What is the importance about the epimysium, perimysium, and endomysium? Is that each endomysium, each perimysium, and each epimysium are going to prolong his or elongate his membrane, and they are going to come together. So all the endomysiums are here. All the perimysiums are here. The epimysium is here, forming what? The tendon. So it's going to be fiber by fiber, and this tendon is going to be attached to the bone. So that's why when you have, for example, a high performance athlete, they're going to have a rupture of the tendon. They're going to heal. They're going to produce a scar tissue. This scar tissue is not elastic, I suppose. And all these connections are being lost. They're going to heal. Yeah, they're going to put it together. But the elasticity of the tendon is going to be different. It's not going to be the same as before. That's why you can tell a patient who has a rupture of athlete, who have a rupture of tendon, that's it. They are not going to improve the performance more than he did or she did in the past because of that. Okay, you got it?
Got it.
Excellent. All right, so let's talk about, I'm going to go to this graphic. Okay. First of all, I want just to draw a cell here, very simple. And we are going to talk about the structures of the muscle cell, of the fiber. Number one, what I want you to remember is that we have striations. I'm going to draw striations like this, in more detail. Of course, there's many more, right? Striations. striations, 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 striations. What are those striations? And then we have another striation here that actually, when you see in the distance, they look like lines like this, but they're actually like this. They're going to be like this. That is called sarcomere. We don't care. All this only is a sarcomere. We don't care about the sarcomere. What I want you to remember is this. This yellow line is a protein. And this protein is called the actin. Actin. Try to nail it. Try to memorize this right now. Actin. Actin. That is a thin layer. It's a thin molecule. It's a thin protein. We call it thin protein. Then we have the red one. The red one are going to be another protein. And this protein is called the myosin. Very important. Myo, muscle, right? Myosin, that is the thick protein. All right. So how is going to be the contraction happen? How is the contraction going to occur? Look at this. These are, this is a line, like a line, but it's not a line. It's going to be a group of molecules like this. A group of molecules that are actually the proteins. Proteins, right? The proteins. These actually I just draw it as a line. So what is the characteristic of the myosin? This is the myosin, and the other one is the actin. Look at this. They are going to have the distribution are going to be like this. They're going to create like hooks. Look at this. Look at that direction of the hooks. Look at that direction of the hooks. Look at that direction of the hooks. It's like going to create like hooks. Like this. In that direction, like this. All right, so we have here going here more and more and more and more. All right, so let's see only that enough. And then we have this is the actin now that is in yellow, is the actin are going to have the hooks in which direction? Like this. See? In the opposite direction. They're going to be like that. So if you see here, it's like going to be two hooks that are opposite. They are going to clinch each other, right? So you have one hook like this and the other hook like that. And what they are going to do, there's many hooks that are going to do this. They're going to just get attached to each other. And that's going to decrease the sarcomere, making the whole cell contract. And that is going to be the contraction. That is the contraction. Then what happens? When you have the relaxation, the hooks are going to do this. Open, and then return to his previous place. So who are doing that? Are these proteins that are the actin and the myosin. The thick protein, myosin, and the thin protein, the actin. You okay with that?
Yes.
Okay. So just keep in your mind, we have the myosin and the actin. Okay. There's other proteins that I'm going to tell you in a few moments. All right. So now what I'm going to do is the next step is to learn how is the structure of the cell membrane of the muscle. I'm going to make this square here to make a close-up. And that close-up is going to be here. Okay, so this is the cell membrane, all what is in purple here. This is the cell membrane. Here is inside the cytoplasm. So if I have a cell here, I'm going to make a close-up here, and that is what I'm looking here. Okay, all right, so number one. Here we have the cell membrane. This is the cell membrane, and I want just to tell you one of the characteristics of this muscle is this. The muscles are not going to, the cell membrane is not going to be continuous like this, exactly or precisely like this. It's going to be most likely like this. Look at this. This is the cell membrane, and then they're going to go in and up again. It's going to continue. It's going to be in. So that is the cell membrane. The cell membrane is going to have that configuration, like this, in and out. So they are going to go here. So no, in and out is deeper and superficial. And that is how the the fiber of the muscle are going to be. So that are going to be, look at this. This is like a roller coaster, right? The cell membrane coming, traveling, all the way down, and then all the way up. So that is what we call, look at this. This is kind of a shape of a T. If you see, it's like a T. So that's why these are called the T tubules. These are the T tubules. This is the T tubule. So you are the T tubule. So that is coming in, coming down, and going up, and continue the cell membrane to the next. So that is the T tubule. Very important to actually know how the muscles are going to work. Okay? All right. So that is the T tubule. Now, here we have, we already talked about the cell membrane, the T tubule. We have here coming down, and this basically looks like a Golgi apparatus, something like that, or endoplasmatic reticulum. All this structure, this in blue, all this that is in blue, all this is in blue, that is the sarcoplasmatic membrane, sarcoplasmatic reticulum. Sarcoplasmatic reticulum. Okay, so but you have a question mark in your mind. So, but what about the endoplasmatic reticulum? The difference between sarcoplasmatic and reticulum is that sarcoplasmatic reticulum is a variation, is a specialized type of endoplasmatic reticulum. So, but endoplasmatic reticulum is different from the sarcoplasmatic reticulum. The characteristics of the sarcoplasmatic reticulum is that they are going to deposit and contain calcium, 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 calcium. Calcium is inside of this sarcoplasmatic reticulum. Okay, so that is, we have the T tubules, we have the sarcoplasmatic reticulum, we have the calcium. Where are these calcium? Calcium are inside the sarcoplasmatic reticulum. Calcium is inside the sarcoplasmatic reticulum. Okay? All right. Now, so this is structures that you need to remember. Here we have the actin in blue here, and we have the myosin here. See the hooks, kind of hooks, right? So they're going to be actin and myosin. This actin and myosin. All right. So here we have a troponin, another protein that you need, is the only another protein that you need to remember, but that's it. Troponin. Troponin is going to lock. The actin and the myosin. What does it mean, lock? Lock means when the troponin is present between the actin and the myosin. When the troponin is present between the actin and the myosin, they do not contract. They will not actually hook each other. They are not going to do anything. It's like you have a boyfriend and a girlfriend. They are together, and there's a third party there. All right, they are actually waiting until the friend is leaving in order to be able to hug each other. Okay, so that is basically boyfriend and girlfriend, the actin and the myosin. When the troponin is there, when the friend, the third party is there, actually you don't do anything. You just talk or whatever, right? But as soon they leave, you start to hug each other, your girlfriend or your boyfriend. All right. So that is the troponin and the actin and the myosin. You okay with that?
Yes.
Okay. All right. Next. So that's all. I'm building up the layers. So we are going to put together everything in a few moments. Okay. So now here we have the terminal axon. This terminal axon, like how I can explain. Do you see these machines? Do you see these machines that said, all right, so let's do exercises without efforts. And they put you a machine and they put it in your abdomen. And the machine start to make your muscles of the abdomen start to contract. It's like you are doing exercises, reading your news, your news in the internet, right? So that's what it's telling. Do you see these machines before? Or you heard about that?
The abdo.
Yeah, I've seen those.
Yeah. So how is this machine working? The machine is working because it's sending electrical impulses. That is the proof that the muscles, how they are going to contract, because they receive electrical impulses sending from that nervous system. So these electrical impulses are the one who are going to make the contraction of the muscle. Contraction of the muscle. Okay, so now here we have, this is the terminal axon. So, the nerves are going to have the body, the dendrites, the axon, and we have the terminal axon. So each fiber, each muscle cell are going to have one of these fibers. Every single fiber. So can you tell how much nerves endings are going to be attached to a muscle? It's a lot of them. Okay? So that is actually what happens. And this is one of these. So here we have the terminal axon, and here we have a specialized area. Look at this. This is cytoplasm, a cell membrane. But this cell membrane is different from this area, or no? Right? So this area of the cell membrane of the fiber of the muscle is a specialized area that is actually the junction between the nervous system, nerves, and the muscle. This area that is located here is called the neuromuscular junction. Very simple. So that is the area where every single fiber of the muscle are going to have located. It's the neuromuscular junction, or called the NMJ. The NMJ is where these are going to receive what is sent by the nerves. Now, this nerve are going to produce, as you know, the nerve cell is like a cell. It's a cell, right? A cell have a nucleus, do translation, do transcription, translation, that produce proteins. So the nerve cells produce proteins too. These proteins produced by the nerve are going to be called the neurotransmitters. So what are neurotransmitters? Neurotransmitters are proteins. Produced by whom? By the nerve cell. Neurotransmitters. Which neurotransmitter we have? Nerve cells produce adrenaline, noradrenaline, GABA, glutamate, serotonin, and what we have here is the acetylcholine. So all these granules that you see here are produced by the nerve cell. That is a neurotransmitter. Acetylcholine is a neurotransmitter, and this is going to degranulate. So that means release from the terminal axon into the neuromuscular junction. So this is basically acetylcholine. And who is going to promote the release of acetylcholine is the electrical impulse. The machine that you put in your abdomen to do exercise without effort, that is the same principle. So electrical impulse in the terminal axon are going to release the acetylcholine. The acetylcholine are going to get into the neuromuscular junction, and what is going to happen? What is going to happen is what we call the electrical impulse. These electrical impulse are going to make changes on the charges outside, inside. These electrical impulse are traveling. So it's traveling like this, traveling like this, traveling like this. And when they are traveling like this, they are going to make enter sodium. That is the first thing you need to remember. Sodium is going to get in. Sodium is going to get in. So we have the electrical impulse are going to release the acetylcholine, and is going to be accepted by the NMJ. This produces an electrical impulse that makes sodium enter into the cell. Sodium is going to get into the cell. This electrical impulse then is going to continue through the cell membrane and get into the T-tubules. It's going to get into the T-tubules. It's getting into the T-tubules. In the T-tubules, what it's doing in contact with the sarcoplasmatic reticulum, calcium deposits here are going to basically go out of the sarcoplasmatic reticulum, where? To the cytoplasm. If you see here, the calcium is already in the cytoplasm. Calcium is in the cytoplasm. Now, this calcium, what it's going to do is to tell the troponin. They're going to call by cell phone, and they say, troponin, get out of there. So actin and myosin, they want to be together. So calcium, what is coming, is telling the troponin and remove the troponin, putting away the troponin. Now the actin and the myosin are alone, and they want, what the first thing to do is to hug each other, and that is produce the contraction of the muscle. Okay?
Yes.
Yes.
Okay. So now, for this, I'm going to explain you a few more things. Well, that's it about the muscle contraction. But what I want is just, do you like cats? You like cats? I like cats, right? You know that, right?
Love cat.
Excellent. And this is our cat. Where is our cat? And that is a giant cat. It looks like a very funny, right? So it's too big cat. So my cat, how much is your cat weight? My cat is about 15, 18 pounds. You have a cat? You weigh your cat? And you can see my cat. It's a giant cat. All right, so why I'm talking about giant cat? Are you crazy? No, I'm not crazy. So I'm going to give you a mnemonic. There you are. G means nothing. I is of giant cat. Giant cat. Okay, giant cat. So what is the giant cat telling you? I is the electrical impulse. Okay, the electrical impulse will go to the NMJ and is going to release acetylcholine, A, acetylcholine. The acetylcholine, then what it's going to make is to make enter sodium. Sodium is going to enter into the cell. Electrical impulse, acetylcholine, sodium enter into the cell, and we have going to follow to the T-tubules, the T-tubules, the T-tubules, the T-tubules. Just a moment, please. Okay, so now let's continue. Okay, so and here what is going to happen when the electrical impulse get into the T-tubules are going to release calcium. Calcium is going to be released. And calcium, what is going to do is to take the troponin, troponin. And the troponin are going to release the actin and the myosin, and the actin and the myosin are going to produce the contraction. You okay with that?
Yes.
Okay, so now let's recap here. So what is the first thing is going to happen is the electrical impulse, correct?
Yes.
Electrical impulse. Then what is happening is going to enter the acetylcholine, correct? The ACH. The ACH is going to get into the muscle, and this is going to make enter what? Sodium. And then the electrical impulse go to the T-tubules, and the T-tubules are going to basically what is going to interact with the sarcoplasmatic reticulum to release the calcium that is deposited in the sarcoplasmatic reticulum. And this calcium, they go to the cytoplasm to look for the troponin. And calcium, what it's doing is to remove the troponin from the actin and the myosin. And the actin and the myosin, now they can actually get together, that contraction. Okay? All right. So please, everyone now. Everyone now. Tell me, what is the I?
The electrical impulse.
The electrical impulse. The electrical impulse. Then this electrical impulse, what is going to release?
Acetylcholine.
Acetylcholine. All right, the acetylcholine. So the acetylcholine, what is going to do after that? What is going to happen with the acetylcholine?
Sodium enters the cell.
Sodium are going to enter into the cell. This electrically, okay, so how to wait to say. So sodium enter into the cell, then the electrical impulse go to the T tubules, and in the T tubules, what are going to is to release calcium. So giant cat. I, electrical impulse. A, acetylcholine. N, sodium. T, giant. T, T tubules. Cat. Ca, calcium is released. T, calcium is taking the troponin and actually release the actin and the myosin to produce the contraction. Is that clear?
Yes, sir.
All right, so listen to this. Every time I ask you to go to, when you go to the bathroom, I told you to put me ACH. ACH is the acetylcholine. The acetylcholine is going to make contraction of the urinary bladder. And that is what is going to make you pee. That's why I've been telling you that when you go to the bathroom, put me ACH. And now it's going to be easier to remember. Okay? You got it? So relate this. Acetylcholine, muscle contraction. Okay? All right. So one more time. Give me an I. Everybody, please.
Electrical impulse.
Give me an A.
Acetylcholine.
Give me an N.
Sodium.
Sodium. Give me a T.
Which one? Any?
The first T.
Troponin.
Tubule.
The T tubule is first.
Tubules. Just kidding.
The T tubule. So just look, just try to follow the graphic that we saw previously. The T tubule, what is going to do? What is happening later? What is happening now?
Release calcium.
Release calcium. And calcium are going to take away whom? The
Troponin.
Troponin.
And the
Tropomyosin.
no. Tropomyosin, don't go to tropomyosin. Troponin only. Troponin. The troponin is, you have, for example, Mark, you have somebody have a boyfriend or girlfriend, right? And somebody's coming, right? Somebody's coming, and you cannot do anything. And that third person is bothering you because you want to go away. Why? Because you want to hug your boyfriend or your girlfriend, right? So once this guy or girl is leaving instead of three, we have two only. The girlfriend and the boyfriend, they can hug each other. The one who is bothering us is the troponin. The actin and the myosin, they want to hug each other all the time. Once the troponin is going away, the actin and the myosin can hug each other. So who is taking the troponin away? The calcium. Calcium is going to remove the troponin from this group. So now actin and myosin are alone and they can actually hug each other. So one more time. What is I? Electrical impulse. What is A? Is going to be the acetylcholine. What is N? Enter sodium. What is T? The T tubules. And then giant, cat, Ca is going to release calcium. And then calcium take the troponin and release the actin and myosin, and now they can hug each other, make contraction. Okay? All right, so very simple now. So tell me, give me an I, everybody.
Electrical impulse.
Electrical impulse.
Without looking papers, without looking any paper, please. Okay. Okay. So Arby, I'm looking at you. Okay. All right. So we have the what is I is the electrical impulse. What is? Give me an A.
Acetylcholine.
ACH, acetylcholine.
Acetylcholine.
Give me an N.
Sodium.
Release of sodium.
Give me a T.
T-tubules.
T-tubules.
Give me a cat. I mean CA.
Calcium.
Release of calcium.
Give me the T.
Troponin.
Troponin.
Troponin. And then the troponin is taking the calcium is taking away the troponin, and that contraction happen. The actin and myosin is going to get together. Okay. Somebody want to go dance along a solo? Okay. So let's see. Elizabeth, you want to make a solo? Let's do it. You start.
What do you want me to say? Oh, electrical impulse, acetylcholine, sodium, something sodium.
Then
The T-tubules.
then
Calcium.
release, right?
Yeah, calcium release. Sorry.
And then what
the
happens?
troponin, and then the
It's-
tropomyosin.
No, don't mention tropomyosin.
Okay.
Tropomyosin. That's the troponin. Nobody mentioned the tropomyosin. It's not. Okay, it's happening, but it's not important. So what I want you to remember are those, because you're going to do troponin when you are doing heart attack later on. That's why I want you to talk about troponin, to get familiar with that. Okay. All right. So thank you, Elizabeth. Catherine, please, you want to make a solo without looking paper?
Okay, sure. Electrical impulse, and then sodium is released, and then T-tubules.
No. Follow the giant cat.
Okay, sorry. So electrical impulse, acetylcholine, and then sodium, and then T-tubules, and then calcium, and then it removes the troponin, and then the contraction happens.
Excellent. See how easy is that? That is the Christmas tree. And then we can put the decorations. The decoration will be, we are going to see that in a few moments. Decoration will be, for example, the electrical impulse is going to come from the motor neuron. Motor means locomotion, movement. So that neuron that are going to be attached to the fiber is actually a motor neuron. This motor neuron are going to transmit the electrical impulse to release acetylcholine, a neurotransmitter. These neurotransmitters are going to act with a neuromuscular junction, producing the entry of sodium. This sodium then are going to accumulate in the cytoplasmatic membrane, and the electrical impulse continue to the T-tubules. The T-tubules are going to release calcium from the sarcoplasmatic reticulum, and the calcium are going to get rid of the troponin, making the actin and the myosin interact and produce the contraction. Simple way to say, the electrical impulse release acetylcholine, enter sodium, then this electrical impulse goes through the T-tubules. The T-tubules are going to make release calcium from into the cytoplasm, and the calcium remove the troponin to produce the contraction, the actin and the myosin. Simple or not?
Yes.
So just remember the giant cat. We have I. The I is the electrical impulse. A is acetylcholine. N the sodium. T the T-tubule. Ca the calcium. And T the troponin. So in my head I have the mnemonic. Until you get used on that, then you can get rid of the mnemonic. But at the beginning you need to do that. Okay, giant cat. Electrical impulse, A, acetylcholine, N, sodium, D, tubule, Ca, calcium, T, troponin, and that's it. That's all what you have. You have all the muscle contraction in that memory. You okay with that? All right. So please practice that when you are not driving, when you are walking, or when you are doing something, when you're eating, when whatever you are doing, try to review that at least 10 times, and you will see that is going to stick in your mind. This is the first part. We are going to get into more detail, but no in different perspective. And please, if somebody is asking you how do you know all this mechanism, you're just going to say that it's just muscle contraction. You don't say anything else, but don't tell anybody about the giant cat, because there is no giant cat in any book, in any material, because this is actually created by me. So that is the easy way to remember about muscle contraction. At the conclusion of all this, you need to always remember that acetylcholine produces muscle contraction. And I just want you to link the urinary bladder. When you go to pee or poo, you contract the rectum, you contract the urinary bladder. And that is because acetylcholine is coming there. So when you go to pee, acetylcholine. When you poo, acetylcholine. All right. So we are going to just remember that. You okay with that?
Yes, thank you.
Yes.
Yes.
Okay.
Thank you.
Sure. All right. So you want a break? I think we can have a break. Right. So I'm going to see you at 6:36. Okay. All right.
Sounds good. Thank you.
Thank you. Hello. Okay, so let's 1 minute away. Okay, so let's get started, please. Don't miss any second, please. What is coming is very important. Okay, so here that is what we have about the muscle contraction. But now I'm going to draw something that is going to be useful for the rest of your career. Okay? All right, so please pay attention on what I'm going to say. Okay, so number one. Here is what is going to happen in the smooth muscle. So this is the physiology of all what they're going to write down here. Number one, the smooth muscle. That is what is going to happen in the striate muscle. That is going to happen in the cardiac muscle. And that is going to happen in the nerve cell. Okay, so what is going to happen is this. Look at this. I'm going to make a chart that is going to be useful for many pathologies, a lot of pathologies. So balance, electrolyte balance, kidney problems, heart failure, arrhythmias, hypertension. They're going to help you in all this stuff, and that's all huge. So let's start. So I'm going to draw here an axis. Let me see if I can make it straight lines here. And here we have another axis. Okay, so now this is what you have here inside these two lines are going to be inside the cell. That is going to be inside the cell. And everything that is outside is going to be outside of the cell. Outside of the cell. All right. Of the cell. Inside the cell. All right. So now I'm going to graphic something that is going to weird probably, but you will understand very soon. Let me see if I can do straight lines. Okay, and then I'm going to draw the straight line here. There you are. Okay, so. All right, so this is what is happening inside the cell. Listen, please. This is what is happening inside the cell. Okay, so in this first part are going to, this line, can you see this line, vertical line? Whatever happened on the left, what happened all this here is going to be the contraction of the muscle. Contraction of the muscle. Contraction of what? Of the smooth muscle, striate muscle, cardiac muscle. We are going to leave at the end the nerve cell. And everything that happened on the right side of this vertical line is going to be the relaxation. The relaxation. The relaxation. So when the contraction is going to occur in the cell, they are going to enter. What is going to enter first? Somebody can tell me?
Sodium.
Sodium. Excellent. Sodium. Sodium starts to get into the cell. This is inside the cell. So sodium starts to get in, and the sodium starts to accumulate, increasing concentration inside of the cell of the muscle. and they are going to get to this tip. In order to get to the top, they are going to enter another electrolyte. And who is that?
The teeth.
Potassium? Oh, no,
Calcium.
calcium.
It's going to happen calcium. Calcium is going to get in. So now, actually, that is very simple. So sodium start to enter, accumulate, increase this line, concentration of sodium. And then when this happening is when they're going to complete the contraction, they are going to enter the calcium. Calcium is getting inside. Now, from this, what happened? Down the hill. And down the hill, we have other elements that are going to go out. So potassium is one. Potassium is going out. Then are going to be the chloride. Chloride is going out. And the magnesium are going out. So those are the 5 that you need to remember. So potassium, chloro, magnesium are going to go out of the cell. So when sodium and calcium are going to complete the contraction, the relaxation is going to occur. And for that, potassium, chloro, magnesium are going out of the cell. You okay with that?
Yes.
Excellent. Now, talking about the nerve cell. The nerve cell are going to produce the electrical impulse. This electrical impulse of the nerve are going to be called the depolarization. Depolarization. This depolarization is the electrical impulse. Is the electrical impulse. Electrical impulse. I'm going to put electrical impulse. All right, so when the nerve cell are going to produce the electrical impulse, what it's going to do is to make enter sodium. Sodium, enter. And when they produce the electrical impulse, it's going to enter the calcium. And then when down the hill, the nerve are going, after they produce the electrical impulse, there is a time of rest. There is a time that they are not going to produce electrical impulse. So that is the time because we have electrical impulse, it's like a pulse, are going to be electrical impulse, silent. Electrical impulse, silent. Electrical impulse, silent. So electrical impulse are coming by pulses, not as a continuous electricity. It's going to be by pulses. And when that time is happening, it's going to be called the repolarization. The repolarization. The repolarization is the rest stage of the cell. Rest stage of the cell. The depolarization and repolarization are going to be called the action potential. Action potential. Action potential. What is action potential? Potential is the formation of the electrical impulse. And this formation of electrical impulse are divided in the beginning, the depolarization and the repolarization. Okay? All right. Let's make the applications about this. All right. So, for example, I will tell you something. When you have, let's run 20 miles. 20 miles. 20 miles. When we run 20 miles, you are sweating a lot. Yes or no? And when you sweat, you are going to lose sodium. Proof of that, taste your sweat. Your sweat is salty. You're losing sodium. Other causes of loss of these electrolytes will be, for example, the dehydration. When you are sweating, you are dehydrated. Or you cannot drink water, you are dehydrated. Or, for example, you have too much vomits, or you have too much diarrheas. When you have too much diarrheas or too much vomiting or bleeding, you are bleeding, you don't have enough sodium, you don't have enough calcium. So what happened? The electrical impulse are not going to occur. The electrical impulse are going to be very, 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 very difficult to be produced. Question about that. So everything that you're doing right now, you're listening to me, you are actually using electrical impulses. What you are looking at in the screen, you are using electrical impulses. When you are thinking, you are concentrating, you are using electrical impulses. when you are focusing, trying to focusing, you have electrical impulses. When you are doing whatever, you're moving your right hand or your left hand, or you're moving your head to the right or left, you need electrical impulses, because these electrical impulses are going to stimulate the muscles too in order to make action. Now, if you're losing these electrolytes, what is going to happen? You cannot focus, you cannot think, you cannot concentrate, you cannot move. So remember a person who are totally dehydrated, or low levels of this sodium and calcium. They are not going to be able to do anything. They are totally, basically resting because they are weak. Why they are weak? They are weak because they don't have sodium and calcium. For example, do you see tennis? When you see a tennis game, there is some games that last 4 hours, 6 hours. What they are doing, the tennis player are going to eat banana. What they eat banana? Why? Because they have potassium, basically. And potassium is not in the right proportion. So they can actually produce no relaxation. And what happened with this tennis player? They can have cramps. So they eat banana to prevent cramps, right? Another thing, for example, the cardiac muscle. The cardiac muscle, you have looped up. Looped up is the muscle, the heart is contracting, systole and diastole, right? Contract, looped up. That is the rate between 60 to 100. But suddenly you have a heart rate like this, looped up. That is arrhythmia, tachycardia. So what we are going to do? What about what happened is that you have tachycardia because there's a lot of sodium getting in and calcium too, and actually produce the contraction. What about if I have a medication who are going to diminish the entry of sodium? No blockage completely, but they are going to diminish the entry of sodium. Instead to be looped up, now you're going to have looped up. And the same, there is medication for calcium. Calcium, if you block the entry of calcium, they are going to basically diminish the heart rate, the calcium. Now, calcium, for example, you have here the artery, that is a normal artery, and the normal artery produces basal constriction. Basal constriction. I told you that when there is basal constriction, the blood is very difficult to flow on that area. So actually the heart needs to push harder, increasing the blood pressure. So what about if blood pressure? What about if I give some calcium channel blockers, that are going to basically relax the muscle? Instead of this, are going to produce vasodilation, and the blood pressure are going down. So all these mechanisms are going to be useful to remember for all these pathologies that we are going to see later on. And if you remember this chart, the names of different medications are going to be so easy for you to remember. So just remember, so what is going to enter first is the sodium. What is going to enter second? The calcium. And that is produced in the muscles, the muscle contraction. But in the nerves, produce the depolarization. And then when down the hill, we have the exit of calcium, chloride, and magnesium. They are going to produce the relaxation of the muscles, and they are going to produce in the muscle, in the cell, the repolarization. Okay? For example, do you see in the over-the-counter on the Walgreens or something, magnesium? Magnesium?
Yes.
Right? And what is doing magnesium? Magnesium is going to calm you down, right? Right? It's calming you down, right? It's going to make you to relax. Yes or no? So you want relaxation, you take magnesium. Magnesium are so high in the blood, the magnesium cannot be pushed out because it's too much in the blood. Magnesium is still longer time in the cell. And this magnesium, what it's doing is to make longer the relaxation and make longer the repolarization. And that is what is doing the magnesium. Okay? So potassium, for example. Potassium, when you have high levels of potassium, you're going to have arrhythmias. You can have bradycardia or you have tachycardia. So, and chloride, the same thing, is going to be always associated with the potassium. So this is the chart that I want you to remember forever. So you have all this smooth muscle. Smooth, for example, in the, let's put a kidney stone. Kidney stone in the ureter, the kidney, the stone is going to be nailed on the ureter. So what we need to do is to basically dilate the ureter in order to make the stone pass through. And what we are going to do is to relax the muscle of the ureter. How we are going to do? With calcium channel blockers, for example, okay, or to give magnesium. So all of these are going to contribute for the treatment of the disease. So that's why I want you to remember and keep it for you forever that smooth muscle, striate muscle, cardiac muscle, and the nerve cells are going to work in that way. Always the same. These graphics you see here are going to be variable. In the nerves are going to be more spike like this. In the heart are going to be something like that. In the muscle are going to be something similar like this. But the concept is the same. Okay? We got it?
Yes.
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 So what is going to enter first? Sodium. What is enter second? The calcium. That is the contraction in the muscles or the depolarization on the nerve cell. Then when you have the muscle contraction, muscle relaxation, what is coming out? Out of the cell, potassium, chloromagnesium. Potassium, chloromagnesium. Potassium, chloromagnesium. That's why electrolytes are so important, because that is going to make work basically the whole body, all your muscles and your nerve cells. Okay, we got it?
Got it. Thank you.
Excellent. All right, so let's keep moving. All right, so we have the filaments that are proteins. Thick protein is the myosin. Question for the exam. Thin protein are going to be the actin and the troponin. Just remember the actin is the thinnest one. Okay? Forget about the tropomyosin. I want you to remember the actin and the troponin. Then we have the neuromuscular junction. Where is the neuromuscular junction? Is in every single cell of the muscle. This neuromuscular junction, they have the receptors for the acetylcholine. The acetylcholine are going to look for these receptors. Here we have the nerve with the terminal axon releasing the acetylcholine, and the acetylcholine are going to look those receptors. Where are located these receptors? Are located in the neuromuscular junction. Neuromuscular junction. And this neuromuscular junction receive the acetylcholine, and this reaction, these are going to make actually the depolarization and release the sodium that is going to enter into the cell. These action are going to make the calcium go out from the sarcoplasmatic reticulum and go act into the troponin to produce the muscle contraction. All right. All right, so let's do this video that I was looking for that. I didn't see the other video in the previous. But let's do this here. Okay, everybody's ready?
Drinking water before bed burns 46 pounds in 2 weeks.
No, that is not.
If you are struggling to lose weight and you're...
Okay, all right.
The 12
Listen.
steps to muscle contractions.
In this case, we are talking about the heart. It's a muscle. So step number 1.
Step 1. Neuron action potential arrives at the end of motor neuron.
Focus on the motor neuron. The motor neuron. The motor neuron is the nerve that is going to get into the neuromuscular junction. This is the motor neuron. They go to the muscle, neuromuscular junction, and produce that contraction. That is the electrical impulse. This graphic is familiar? Yes. Look at this. Electrical impulse coming, electrical impulse, motor neuron are going to produce the actually depolarization, releasing the acetylcholine. Okay, so this is the changes of charges, is the depolarization.
Step 2, acetylcholine is released.
That is the terminal axon, and they are going to, these granules are producing the degranulation. Here in green are the receptors for acetylcholine. Acetylcholine. The acetylcholine get into the receptors of.
Step 3, acetylcholine binds to receptors on motor end plates.
So they're going to link into, and what happened now?
Step 4, sodium ions rush into muscle fibers.
There you are. So they are going to get all these. This is outside and getting through the gates that are going to get sodium into the cell muscle. All the reds are going to come in. That's the sodium.
Step 5, muscle action potential sweeps into T-tubules.
So that these invaginations that you see here, they are going to be the T-tubules. This is the surface of the cell membrane. That is of the cell membrane of the muscle.
Step 6, sarcoplasmic reticulum releases calcium.
So all this snow that you see here is because they're because of these.
Step 7, calcium binds to troponin.
Okay.
Step 8, when calcium binds to troponin, it causes tropomyosin to shift and expose myosin binding sites.
And they are going to produce the contraction of the muscle.
Step 9, myosin binds to actin.
Those are the hooks I
Step
was
10,
saying.
myosin pivots, pulling actin filament.
They're going to pull the actin.
Step 11, myosin releases from actin. Step 12, myosin re-extends into ready position.
All right, so that is what happened in the muscle contraction. In the muscle relaxation, what is going to occur is that the calcium are going to return to the sarcoplasmatic reticulum. So, and the troponin is going to get attached to the actin and the myosin, and that is the relaxation. So the contraction will be the calcium is telling the troponin go away. And when they go away, actin and myosin are going to contract. That is the contraction. Now, if you want to have the relaxation, what it's going to do is the calcium go back to the sarcoplasmatic reticulum. So the calcium are not going to be able to get rid of the troponin. So troponin get attached to the actin and the myosin, and the contraction is not going to occur. It's the relaxation. Simple or not? Okay? All right. So now let's see. I'm curious about the other video. I don't know what is the video. Hopefully something. All right, so let's see. I didn't check the video in the morning, but I'm going to see what's giving me. Video and VA. What probably is that? Go to home. No. Okay. All right. So that is about. So any comment, please? Chelsea, tell me. What do you think?
I thought you explained it really well. It's easy to memorize with the steps.
Excellent. Thank you so much. Okay, Mark, you okay with that?
Yes, perfectly fine.
Excellent. All right, so let's continue. Let's make, you already have the Christmas tree. Now let's do our what? Our decorations. All right, so now the muscle. Muscle are going to need oxygen. That is by 100% oxygen. Just remember, when you have in the cell, in the muscle cell, cardiac muscle, etc., are going to have the glycolysis, right? Oh, sorry, what is that? The glycolysis. The glycolysis produce the pyruvic acid. Pyruvic acid go into acetyl-CoA. The acetyl-CoA go into the mitochondria. And there where you need a lot of oxygen to produce the ATPs, right? So muscles are going to need oxygen all the time. And who is going to contribute to that is the myoglobin. Myoglobin are going to actually capture oxygen. Look at this. Myoglobin is the word similar to the hemoglobin. Hemoglobin is in the red blood cells. Hemoglobin action is to capture oxygen and carbon dioxide, right? But we are focusing oxygen right now. The myoglobin is similar to the hemoglobin, but this myoglobin belongs only to the muscle. So in the muscle, we have this protein. It's a protein. Myoglobin, hemoglobin are proteins. The hemoglobin in the red blood, in the blood, and the myoglobin inside the muscle cell. These myoglobin have the ability to capture oxygen. Why? Because your muscle needs a lot of oxygen. So in addition to that, we need glucose. You cannot do exercises without glucose. So the glucose is the source of energy, right? Easy energy, glucose. So the glucose are going to be entered into the cell to produce all this effect. But one thing you need to remember is that the muscles, we have the liver and muscles is where mostly of the glycogen is going to be located. Glycogen. What is glycogen doing? What is glycogen? Glycogen is a polysaccharide that is composed by hundreds and hundreds and hundreds numbers of glucose. Glucose, a lot of glucose together that form a molecule called the glycogen. The glycogen are going to turn into glucose, as you already know. Glucose. Okay, glucose. When they turn into glycogen into glucose, that is going to be called the glycogenesis. And the glucose can turn into glycogen. The opposite. It's the opposite. Glycogen into glucose is going to be the glycogenolysis. And the glucose into glucose into glycogen is going to be called the glycogenesis. These are very important concepts. When you are actually low on sugars, the glycogen that is located, basically glycogen, located basically in the liver and the muscles, they're going to start having the glycogenolysis that produce glycogen into glucose for a source of energy. We okay with that? And you remember
Yes.
the glycogen, thank you. The glycogen is how much glycogen you can deposit in your body. You can deposit 2 pounds of glycogen, 2 pounds of glycogen, 2 pounds of glycogen distributed into the liver and the muscles. This glycogen, when you are in starving situations, you're going to use it and start to be active in the first 12 hours. And it's going to last, the last molecules of glucose coming from glycogen is going to last up to 7 to 12 days. 7 to 10 days. All right, so that is the concept here is what is inside the muscle cell. We have the myoglobin that is going to help to capture oxygen for the muscle activity, the glycogen that is the storage of energy in the form of carbohydrates in the muscles, and we have the creatine phosphatase. The creatine and phosphatase, I'm going to put it here. It's a mistake there. A creatine phosphatase. Phosphatase. Okay, phosphatase. Creatine phosphatase, you see that ASE is an enzyme. So this is crucial. This is so important, very important. So please make priority in this creatine phosphatase. Creatine phosphatase is an indicator of the muscle. What does that mean? So you know that the muscle are going to contain the creatine phosphatase here in the enzyme. Where is that? Inside the muscle cell. When you destroy the muscle, for whatever reason, there is medications who produce damage of the muscle already. They are going to, these enzymes are going to escape. They are going to escape, and they are going to go into the blood. So when you take a laboratory test of creatine phosphatase, you will see that this is being elevated, and that means an increase of muscle damage. So all this, when you are, this is very important. Why? Because they can indicate kidney failure. Why is that? When you destroy muscles in huge number, all these myoglobin, all these proteins like troponin, the myosin, and the actin are going to be totally released into the bloodstream, making the blood very thick, very thick. At the time they are going to pass through the kidneys, the filtration is going to be blocked like a mud, M-U-D, like a mud. And these mud are going to make the kidney fail. And that is caused by, actually, how you can detect that with the creatine phosphatase. I will give you one example. For example, there's some people who use Lipitor, that is for decrease the cholesterol. This Lipitor side effect will be to destroy cells of the muscle, and you can die with that. So you need to check with a creatine phosphatase elevation. If you check the creatine phosphatase elevation, that is going to give you an idea if you need to stop or continue the medication. Another example, do you hear some people in the news, I don't know, in the past, whenever, that person was beaten to death. So beaten to death. So they was smashing the muscles one and again and again and again. The muscles, so massive destruction of the muscle that all these proteins coming out from the cells, and they are going to go to the kidney and produce kidney failure. And the patient can die in 8 hours. Do you hear this in the past somewhere, that was beating until he died or she died? Right?
Yeah.
That is the explanation. Okay, we got it?
Yes.
Okay. All right. So basically we have oxygen consumption. This is important too. Let's go to the gym. You go to the gym, you're too excited, you have your new tennis shoes, you have your new everything, your bag, everything, you are ready to do the Mr. California or Miss California, right? So you go and you want to do in one day everything that you didn't do for many years. So actually you say, oh, you're happy after that, right? You go home, you go to sleep, and in the morning you open your eyes, and the only thing that you can move is your eye. You open your eyes, because your muscles are in extreme pain. Okay, so that happened to you or no? That happened to me.
Yep.
So why is that? Why is that? Because basically the coach in the gym is telling you need to come 3 times a week. Oh, they're greedy. I want to come every day. No, they tell you the right thing. Come 2 to 3 times a week. Why is that? All right. And the coach is telling you in every exercise you are doing, you need to take a lot of oxygen. Contraction. Inhale, contraction, right? So that is the way to do exercise. Why? Because you are providing under physical activity more oxygen. And why is that? So they give you oxygen because the acetyl-CoA, I didn't say acetylcholine, the acetyl-CoA in the Krebs cycle, they need oxygen. Now, if you don't provide enough oxygen, and you're not being trained in the past, you cannot oxygenate your body if you were trained. When you are trained, you have a lot of oxygen, but when you are not, you don't have enough oxygen. So what happened? The body, you force your body to do exercises. And what happened? You cannot produce ATP because you don't have enough oxygen in the Krebs cycle. So what is doing the body? You still need ATPs. So they are going to do the anaerobic reaction, that is the glycolysis. The glycolysis is going to give you only 2 ATPs versus 36 ATPs of the Krebs cycle. But these 2 ATPs is better than nothing. So that's why you are still doing this anaerobic reaction to produce at least 2 ATPs. But what is the problem here? The problem here is this. The problem is when you are doing this situation without enough oxygen, the pyruvic acid accumulates so much into the cytoplasm that are going to turn into lactic acid. This lactic acid is the one who accumulate in the muscle cell, and that is what produces the pain. That is what causes the sore of the muscle. And how long is going to take to be absorbed this lactic acid again into glucose is going to take about 2 to 3 days. 2 to 3 days. 2 to 3 days. So that's why the coach is telling you better come 2 to 3 times a week. Why? Because in between you need to recover from the pain of the exhausting exercise. Is that clear?
Yes.
Yes.
So now when you go into the gym, you know what to expect. Okay? By the way, I have a gym in my house, so I would suggest not to have a gym at all in your home because you're not doing it. A gym in the house is just decoration right now, in the last 2 years, for at least. Okay? All right. Any question, please?
None so far.
Okay. All right. So let's go a little bit, change a little gears in just to mention, because that is part of the lecture, of the syllabus. I'm going to talk about the isotonic and isometric contraction. This is the isotonic.
Dr. G?
Yes.
Sorry to interrupt, just to keep the track of time. What time would you like us to go on our lunch break?
7:25.
At 25? Okay, sounds good.
Okay, excellent. So what time is it now?
It's 7:11.
Oh, 7:11. There you are. 7:11. All right. All right. So let's talk about the muscle contraction, and we have two types of actions. We have isotonic and isometric. Okay, isotonic is not like osmosis. Okay, it's different. Okay, so isotonic. Isotonic, you have a weight in your hand of 10 pounds. 10 pounds. Whatever position you are going to have, you have the same weight. Okay, the same weight. So you have the same tension, the same weight, but changing the length of your muscle. So when you are doing exercises with 10 pounds, how you call that? 10 pounds weight? 10 pounds. 1, 2, 3, 4, 3. So I want you to try, well, anyhow, another time. All right, so we have, that is the isotonic. Iso means equal. Equal what? Equal weight, right? Same tension. Isotonic. Tonic means the stretching out of the muscle. That is stretching out. Tonic. Iso means equal. All right, so you have the same weight, and you actually change the length of the muscle, the biceps. That is isotonic. Isometric is when you push the wall. So actually you have the same length because you are pushing the wall and the same tension. For example, you try to open the door, push the door, or you push with your hands the table like this. You are doing tension, but without changing the length of your muscle. That is the isometric. The one who are going to use more oxygen are going to be the isotonic. Okay? Then we have muscles that are going to work together. The muscles who are working together are going to be the prime movers and the antagonists. All right, so the prime movers are going to be, example, the biceps, your biceps. Your biceps are going to be the prime movers. So the antagonist is the one who is doing the opposite. We have on the top of your arm here, we have the biceps, and behind we have the triceps. This is the triceps. Here's the triceps. Now, if you contract the biceps, this muscle below, they need to stretch out. So that's why it's called opposite. They're going to be antagonics. Now, if you're going to extend your upper extremity, if you extend, you need to relax the biceps by the triceps, but the triceps is going to contract. Those are actually the antagonists. Okay? the prime movers and antagonists. What are best example? The biceps and the triceps. The synergistic or synergist muscles are going to be secondary muscles. For example, below or under the biceps, we have the brachial muscle, the brachiobrachialis, the brachial muscle. So below, under the biceps. So this brachialis muscle are going to contract together with the biceps, trying or helping the biceps to do the function. So those are secondary muscles, and those are going to be basically helpers. Okay? All right. So we are going to mention that. I'm not going to ask that in the quiz, but definitely you need to, this is part of the curriculum I need to go over. All right. So let's talk about any questions so far, please?
None so far.
Okay. All right, so we have the orbicularis oris. Orbicularis oris, where is the oris? Here. Orbicularis oris. Orbicularis oris is the kiss muscle. So you do the changes of your lips in order to make a kiss. That is orbicularis oris. Oris means oral, means mouth. We have the orbicularis oculi. The orbicularis oculi is when you open and close your eyes. Open and close your eye. Oculi. Oculi means eyes. Okay. Orbicularis. Orbi is around. Around. Orbicularis is like muscles that are around. If you see here the orbicularis oculi and the orbicularis oris. Then we have the masseter. The masseter muscle, write down that please, question for exam, are going to be doing the mastication, chewing, mastication. What we are going to do in the break, you're going to say masseter, 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 masseter. That is the mastication. Another important muscle is the sternocleidomastoid, or called SCM, the sternocleidomastoid. What is the sternocleidomastoid? Sterno means sternum, right? The sternum. Cleido means clavicle. Clavicle. What does it mean? That muscle are going to originate from the sternum and from the clavicle, like this. And they are going to go to the mastoid process. Where is the mastoid process? Here in the occipital bone. So you see my muscle here? Can you see my muscle when I move my head like this? Can you see this line? Check it out. Do this
Yes.
in your head and follow the muscle. Follow the muscle. Go deeper. See where they go? They go to the mastoid process. So that insertion of the muscle start in the sternum. Another insertion have in the what? In the clavicle, and then they go to the occipital bone. What area? That area of the occipital bone is called the mastoid process. What is the importance about the sternocleidomastoid? The importance of this muscle is that you are going to say no, yes, you're going to say, you're going to look around here. That is the sternocleidomastoid. The sternocleidomastoid. Is that clear? Movement of the neck, sternocleidomastoid. We okay with that?
Yes.
Yes.
Okay, deltoid muscle. The deltoid muscle is on the shoulder. Deltoid muscle is doing this. So I'm going to contract the deltoid muscle. Relax the deltoid muscle. Deltoid muscle contraction. See? It's going to elevate the upper extremity. Abduction. This is another time. But just remember that deltoid muscle. Why is important deltoid muscle? Because it's a big, strong muscle that we use for intramuscular injections. They're going to have intramuscular injections. We have the pectoralis major. That is on the chest. Okay. We have pectoralis major, pectoralis minor. Just pectoralis is on the what's called the chest muscle, right? So the pectoralis major. Okay, we have pectoralis major. Under that pectoralis major, we have the pectoralis minor. Sometimes it's important to know that because when somebody have cancer or have implantation or aesthetic surgery, they want to implant breast. And the breast is going to be below or under the pectoralis major and anterior to the pectoralis minor. Okay? All right. So we have the biceps brachialis that is going to produce the, as you can tell, the flexion of the muscle. The flexion of the muscle. Another muscles that I want you to remember are the adductors of the thigh. The adductors of the thigh. Okay? We have the gracilis, we have the adductors longus, adductors brevis, and we have the pectineus muscle. We don't care about that. Adductor muscle. What are doing the adductors? So when you have this machine between your legs, they are going to make pressure against laterally, right? So what you're going to do is to push your legs to the middle, to the center, like this. What you are using are your adductors. Very common use in the gym for girls. Girls, they like to have good adductors. So they are going to do this type of exercises. Other way is when you put your leg to the middle, to the center. That is going to be the adductor muscle. Okay, adductor muscle. Just the group of adductor muscle. That is medially here, medially. So you press against weight. You're going to try to close your legs like this. That is going to make the exercises of the adductor muscles. Yes, and here anteriorly we have the quadriceps femoris. Quadriceps femoris go anterior and lateral. So basically, is one of the the largest muscle of the lower extremity. Then posteriorly, we are going to have the hamstring. The hamstring. The hamstring is going to be these three names. You don't care about that, but this is basically posteriorly to the thigh. Posterior to the thigh. So those are the three groups of muscle that we have on the leg, in the leg. The hamstring, the adductors, and the quadriceps femoris. All right, so let's keep going. Here we have, I forget about to mention the trapezius. Trapezius is when you look in the sky, like this, oh, okay. Or when we don't know something, we look at like that, you're using the trapezius. Trapezius is going to be used for climbing, especially when you are doing this climbing rocks, right? So you're using a lot of trapezium. Trapezium is very strong on that. They need to be trapezium to climb, okay? All right, so here we have the rectus abdominis. The rectus abdominis is all this. The rectus abdominis is what we call the 6 packs. The rectus abdominis. Some people have only 1 pack, doesn't care, all right? So, but 6 packs actually is what we have group of muscles, 3 group of muscles, right? This is already the pelvis. That's 1, 2, and 3. So those are on the other side, 1, 2, 3, the 6 packs, okay? Okay, so here we have the gluteus maximus, This is the gluteus maximus, and the gluteus maximus is where you sit down, right? And the gluteus maximus, is important because it's the largest muscle in the body, in the whole body. From head to toe, the gluteus maximus is the muscle, the largest muscle in the body. The largest muscle in the body, the gluteus maximus. Some reference about Enkles and Hesse is kids less than 14 years old. They are not developing yet the gluteus maximus. They don't have a big gluteus maximus. Why? Because age, they are not developed yet. And why is important that? Because intramuscular injections. You cannot give intramuscular injections in less than 14 years old if in addition, the gluteus maximus is not developed, because what you're going to go directly to the bone. And what they're going to do is lateral. They're going to use another gluteus maximum, another gluteus is the gluteus medium, that are going to be lateral to the plane of the patient. All right, so what else here? Oh, on the back. On the back, we have the latissimus dorsi. Latissimus dorsi is the largest muscle on the back. The largest muscle on the back. That is the latissimus dorsi. You go to the gym, you see these guys doing the pulling here, or the like rolling, right? How you call it? Rowing, right? They are going to have the shape of the back like this, like a triangle. So that is the latissimus dorsi. Latissimus dorsi. You okay with that?
Yes.
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 Okay. Now, another muscle I want you to remember is the gastrocnemius muscle. The gastrocnemius muscle. That is called the second heart. The second heart. Why is the second heart? Because here in between, this is one gastrocnemius muscle, and we have another gastrocnemius muscle. There are two of them. In the middle are going to run a vein. When you walk, these muscles are going to contract and squeeze the vein. So the vein is going to eject the blood towards the heart. So that's why the gastrocnemius muscle are going to be called the second heart. Now, the gastrocnemius muscle are going to get attached to a tendon. This tendon is the Achilles tendon. Question for the exam. Achilles tendon. And where they are going to touch? In a bone. What is the name of this bone? The calcaneus. The calcaneus bone. Calcaneus bone. That is on the heel. So that is the Achilles tendon, calcaneus, and we have the gastrocnemius muscle. The gastrocnemius muscle. Okay? All right. So one more thing, the last muscle I'm going to ask you to know is the soleus muscle. The soleus muscle is going to be behind the gastrocnemius muscle. Gastrocnemius muscle. And this is the location of, basically, we have veins there that can be very likely, most commonly located the DVTs, deep vein thrombosis. When you have a thrombus on the veins on the legs, most likely are located in the soleus vessels, because soleus muscle, they have soleus vein and soleus artery. Gastrocnemius muscle, they have gastrocnemius artery, gastrocnemius vein. So it's the soleus muscle that is located under is running the soleus vein, and that is the most common location for clots. Okay? Okay, so let's make a recap here. We have the orbicularis oris. We have the oculi and the orbicularis oris. Here we have the masseter muscle. The masseter muscle. This is the masseter muscle, mastication. This muscle you see here in between is the buccinator. Buccinator is the whistle muscle. You're going to whistle. So you are using your buccinator muscle. We have the orbicularis oris here. Orbicularis oris. The orbicularis oculi. That is around the eye. Right? And that's it about this. Second. All right, so muscles of the neck is the sternocleidomastoid. Sternum, clavicle. Mastoid, part of the bone of the occipital bone. So what they are going to do is to move your head. You're going to rotate the head, of the head, right and left. You're going to bend your neck, flex your neck like this, flex your neck like this. All these movements are going to be caused by the sternocleidomastoid. The other group of muscles that you need to be aware is this muscle. We have the this is the shoulder, this is the scapula, this is the spine of the scapula, this is the humerus, and this is looking from behind. So we have one muscle, the supraspinatus. Supra because it's above the spinous, it's supraspinatus. Infraspinatus because it's below. We have the teres minor, and anterior we have the subscapularis. The subscapularis is anterior. You don't see it here, but all of them are going to be attached to the humerus, and that is the rotator cuff. So do you need to remember this name? No, I don't think so. I will not ask you that. So just think about that rotator cuff. Rotator cuff, just to give you an idea, what is actually when you have a rupture of one of these muscles, that is the movement of the shoulder. Rotator cuff problems is when you cannot wash your back with a soap. So you cannot do this. You cannot do your hand on the back. You cannot do that. So that is very painful, very painful. And the way to calm down the pain is having the arm like this, close to you anteriorly. So that is rotator cuff.
Yeah, I have a what is it called? The subscapularis tear in my rotator cuff.
Oh, really? Yes.
Yeah.
And that happened for many situations, huh? Especially.
I fell.
Okay. It's very painful, right?
I take CBD gummies sometimes to help with the pain because I don't really like taking pills.
Oh, so you're not resolved yet the problem of the rotator cuff?
Well, kind of. I got a bunch of like injections and stuff, but it still hurts from time to time.
Yeah, it looks like your arm is going to fall down, right? Yeah, that's looks like the arm is going to just fall down in pain. All right, so thank you for sharing that, and I'm sorry for that, and hopefully they're going to recover seriously. Honest wishes. Okay, so we have the biceps brachii and we have the triceps. There's different muscles that we have here, but the biceps brachii and the triceps are actually what I want you to remember. Now, the muscles of the respiration. The most important muscle of breathing is the diaphragm. By sure. 100% far away, more than the other muscles. Now, the most important muscle for inhale or inspiration is the diaphragm. So write it down that. Diaphragm, the most important muscle for inspiration. And the intercostal muscle, when you go to Chili's, you buy these ribs, right? What you eat, the muscles are the intercostal muscle. So these intercostal muscles are going to be the expiration or exhalation. That are actually what you must know for by now. Diaphragm, inspiration, intercostal muscle, expiration. Okay, I'm not going to go rectus abdominis. We'll talk about the stuff. I mean the six packs. All right, so and this is going to. All right, so this is the perineum. The perineum. The perineum, we have muscles here that are going to be the elevator anus, etc., etc., etc. So what I want you to remember here is basically the elevator ani. Elevator ani, the below your body, between your legs, that is called the perineum. Perineum. Okay? Here is anterior. This is posterior. Okay? And here we have the orientation. Look at this. We have the anus posteriorly. Then we have here the urethra here. This is the urethra, the exit of the urine. And in between, we have the vaginal canal or birth canal. So this is a must you need to remember. So why I put this picture? Because of the muscles, obviously, but you're going to use a Foley catheter. Foley catheter, they need to drain the urine. So what they need to go, they go here in the urethra. So they go all the urethra down to the urinary bladder, and they put this back to collect the urine. Okay? So why is important to know that? Because there are some people, by mistake, they put the Foley catheter in the anus here. Other people make a mistake and they put it in the vagina. That is wrong. Okay? And the worst, some people think that the clitoris is actually the urethra, and they push, and push. And what it's doing are going to make hurt the patient. All right? So we'll say, but it's obvious how I can make a mistake. You will make the mistake. Why? Because that isn't a normal weight person. What about if you have 300? That is not uncommon to have 300, 400 pounds women. So you don't see anything. You are swimming in fat. So what you need to push, you need actually be sure where you're putting the Foley catheter. And it's not like whatever is going to enter, that is, no. All right, so you need to ask for help on that. So you will see patients with 300, 400 pounds that you don't see anything at all. So, and this, you ask your clinical, there is no uncommon to see this type of mistakes. Okay, we talk about that.
Dr. G, it's 7:35.
Okay, let me finish with these two topics, please. All right, thank you. Strains and sprains. A strain is the stretching or tearing of muscle or tendon. So strain, muscle, tendon. Strain, muscle, and tendon. Sprain is the ligaments. The ligaments. So the ligaments are going, if you have the knee here, this is the knee. This is the knee here. Let's make it like that. This is the knee, the kneecap. Here we have the articulation of the knee. Here we have the muscle. The tendons are going to end here on the bones, but the ligaments are going to be ligaments like rubber bands that are going to basically be on the side to stabilize the articulation. They're going to be like this. Our fibrous connective tissue, when you bend your knee like that, that ligaments are going to rupture, and that is going to produce what we call a sprain. A sprain and a strain. Both are very bad, okay? But when you have a rupture of the ligaments, it's a sprain. When you have a rupture of the tendon, like for example, in the Achilles tendon, rupture are going to be actually some strains. All right, so, and the last thing I want just to go here is the myasthenia gravis. So the myasthenia gravis, look at this, myasthenia gravis is going to be a muscle, and you have the neuromuscular junction here. So this is an autoimmune disorder. Autoimmune disorder is going to be that your own immune cells are going to destroy your own body. That is what happened with the myasthenia gravis, and with the autoimmune disorder. For example, rheumatoid arthritis. That is an autoimmune disorder that is going to destroy the joints, the articulation. All right, so your own body destroy that. This myasthenia gravis, what it's going to do is to destroy the neuromuscular junction, So you need to remember myasthenia gravis, autoimmune disorder, destroy the neuromuscular junction. So you cannot contract your muscle, so you start to have weakness, right? Asthenia, so totally very weak because you cannot contract the muscle. Myasthenia gravis. Okay, all right. So that's it about that, and we are going to get into the next chapter. Any question, please?
None thus far.
No.
Okay. All right, so let's have a break. We are going to have 7:38 is, how much is that? It's 8. We are going to get at 8, correct? All right, so let's go until, my God, I cannot make a summation, right? So it's 40, 22, so yeah, 8:10. Okay. Any questions so far? Do you like a muscular system, muscles? Okay, thank you. All right, I will see you 8:10. Hello. Everybody's welcome. All right, so this is a very heavy topic, okay? So I want your focus and your concentration. Okay, so we are going to use some of the information that we are going to use as a base, the bioscience. Okay, so let's start. Let's get started. All right, so always I will attempt to say this. Can you see my screen?
Yeah.
Okay. All right, so where is coming the word memorizing something by heart? That is coming from the ancient Egypt. That they was thinking that the center of the intelligence, emotions, feelings are going to be located in the heart, within the heart. So since then, they was saying memorizing something by heart, because they was thinking there's 3,000 years or more of this phrase. All right, so let's keep moving. So here we have talking about the head. We don't put here the muscles, but here we have structures like the skull and the nervous system. The skull are going to be formed by the cranial bones and the facial bones. That is actually the skull. Okay, the calvaria is you have these samples in the school that you open the calvaria to see what is inside the cranium, right? So the calvaria is like the small hat, H-A-T, that are located in, so they gather there, and that is called the calvaria. It's composed from some frontal, parietal, occipital, and a piece of the temporal. So that is called the calvaria. And the cranial base, that basically are all the bones that we already know from the cranium, the 8 bones. The facial bones are 14 bones. We are not going to mention about that, because our main topic is to talk about the nervous system. The nervous system, I want you to focus in this area, nervous system, and the anatomy of the nervous system are going to be divided in the central and the peripheral nervous system. Central nervous system is called because all is in the center of the body. So what is in the center of the body in the nervous system is going to be the brain and the spinal cord. We have the brain and the spinal cord. The brain is composed by the brain is not the cerebrum only. The brain is going to be the cerebrum, the cerebellum, and the brain stem. So there is 3 components of the brain. Peripheral nervous system, we will talk about next class. Now, here we have the cerebrum. This is the cerebrum. Here we have the cerebellum. And here we have the brain stem. The brain stem. So all these together are going to be called a brain. So there is a common mistake that they think the brain is only this. It's not. The proper name is called actually the cerebrum. Okay? All right. So saying that, we need to remember about the components of the brain stem. The brain stem, I will say, this is like a flower. A flower is going to be the cerebrum. We have here the stem of the flower, and I mentioned that stem because I don't want you to confuse with the midbrain. People confuse the midbrain, that is the brain stem, is not. So the brain stem is a part of the brain stem. And the flower that is going, the leaf that is on the side will be the cerebellum. The cerebellum. So we have cerebellum, we have the cerebrum, and we have the stem that is the brain stem. Okay? The brain stem is actually the upper portion of the brain stem, pons, and the medulla. All right, so let's start. All right, so the nerve cell, I want just to tell you what is the difference between a nerve versus a nerve cell. A nerve cell is, if you have a rope, you see a rope, the rope, they have a lot of fibers, right? So the rope is the equivalent to say the nerve. And the fibers are going to be the the nerve cells or neurons, nerve cells or neurons. So actually a nerve, for example, the radial nerve, the ulnar nerve, the sciatic nerve, any nerve, the median nerve, all these nerves are actually a group of thousands of even millions of nerve cells that are going to be related to each other, connecting to each other. Okay? All right. So now let's talk about the brain. The brain is going to be the cerebrum, brain, and cerebellum. The cerebrum are going to have the cerebral hemispheres. Hemi means half, spheres means sphere, round. So, and this is what we have here. This is an that is the cerebrum, is divided in two hemispheres. Hemisphere because it's half of one sphere and the other half of another sphere. So that is called the cerebral hemispheres. One question for the exam is going to be that division you see here. This is a line here in the center that is what called a fissure, or that is basically a gap, a space, right? So they're together, they're side by side, but if you put your finger there, you can separate the right from the left hemisphere. You okay with that? Okay. So talking about the lobes. The lobes we are going to talk about the lobes. We have two frontal lobes. Two frontal lobes. We have two occipital lobes. Why? Because of the longitudinal fissure. The longitudinal fissure is going to cut the frontal lobes in two, 1 and 2. Here we have the occipital lobe are going to be divided in 1 and 2. Occipital lobes, or two occipital lobes. We have two parietal lobes. And we have two temporal lobes. I mentioned that because if I'm talking about the bones, I have only one frontal bone. We have one occipital bone. And we have two parietal and two temporal bones. You okay with that?
Yes.
Okay, perfect. So now, to talk about the diencephalon, I'm going to talk about the orange. Let's talk about the orange. So I'm going to explain you in a few easy and make a shortcut, but no sacrifice, not even 1 centimeter of the material. All right, so let's start. Here we have an orange. And the orange, they have a skin. The orange have a skin. The skin of the orange. All right? And here in the center, we are going to have, we have here, right? We have here in the center, not nowadays, but basically natural, we need to have some seeds here. Some seeds. So those are the seeds of the orange, seeds. That is basically what happened in the cerebrum. This portion, the skin of the orange, is the equivalent to, say, the cortex. The cortex, cerebral cortex. I'm going to put cerebral cortex, the cortex. This cortex is made by gray matter. Gray matter. These gray matter are composed by nerve cells, who have the axon there and do not have Schwann cells. They do not have the Schwann cells that contain the myelin. The myelin is a fat, and what is doing the myelin is going to accelerate the electrical impulse. In this case, the gray matter do not have myelin, so it's going to be the unmyelinated nerve cell. All of these are having unmyelinated nerve cells. Then we have the pulp. Here is the pulp of the orange, and the pulp of the orange are going to be formed by the white matter. The white matter. So see, it's gray matter here, cortex, and the pulp is equivalent to, say, the white matter. What is actually the white matter differentiates from the gray matter is that the nerve cells are going to have the Schwann cells. The Schwann cells. These Schwann cells, as you already know, wrapping around of the axon, are going to be a myelinated nerve cell. And that is going to basically increase the speed of the electrical impulse. Why is that? Very simple. Look at this now. Actually, what happened, okay, I will tell you. This myelinated is going to be the speed of, just to give you an idea, the speed of the amyelinated nerve cells is going to be about 1 yard per second, just to talk in football terms. And these myelinated are going to be 100 yards per second. So it's like some of these guys are going to run the 100 yards in 1 second, very fast, right? But the distance in the body is very short. So actually the speed is very, 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 very much remarkable. Now, why is this? The gray matter are going to be, and please listen to this, the skin of the orange is very thin, right? Are going to be the commanders. The commanders. So those are actually like the one who make orders. They make orders. They make orders. So all that decision making that you have every day are going to happen in the cortex. The more cortex, the more gray matter. The more gray matter, more, I would say, cerebral activity, more easy to make decisions, okay? And humans, we have the highest amount of gray matter compared to other mammals or to other animals, okay? Now, these commanders are going to send the orders. The orders to move your hand, to move your arm, to talk, to sing, to do everything that you can imagine. These commanders are going to send the order. So if you send an order, you as a commander, you want to have that your message are going to arrive very fast, right? Very fast, very fast. And for this, they are going to send the orders to the white matter. The white matter are basically the messengers, the one who transmit the order. Transmit the order where? I will see that in a few moments. Now, the lobes that we have, we have the frontal lobe, and this is the summary, so please pay attention. Frontal lobe, the frontal lobe are going to be basically the motor area and the cognitive area. So that is question for the exam. Frontal lobe is the motor area, motor area, locomotion, all the movement of your body. And cognitive, your intelligence, your personality, your emotions, your feelings, right? Your anger, your whatever. So all these, your alertness, you are alert, you are conscious that you are alive. So that is the frontal lobe, the frontal lobe. Then we have the parietal lobe, The parietal lobe are going to be what we call the somatosensory. So everything that you feel in your hand, touch your hand, pinch your ear or your cheek. So all these sensations are going to be actually detected and understood by the parietal lobe. So somebody is pinching me, right? Somebody is touching me. I have pain. So everything is in the parietal lobe, the somatosensory area. I will put here pain, for example, okay? Then we have the temporal lobe. The temporal lobe. We are talking about lobes, okay? The temporal lobe. Temporal lobe, very easy. We have the many functions, right? But we have the limbic system. We have the writing area, the speech, etc. We are going to see that another time. But the simple way I want you to remember is this. Put your finger in your ear. Put your finger in your ear. What you touch here, the ear canal is going to pass the bone. Which bone is going to pass? The temporal bone. And they go to arrive to the temporal lobe. So the temporal lobe are going to be the auditory area. The auditory area. Simple to remember. Auditory area. And the occipital are going to be the vision area. Vision. Please, it's no movement of the eye. Movement of the eye is not. You're not moving the eye because of the occipital lobe. You're actually using the occipital lobe to identify objects, colors. So that is what is all the information coming from outside through the eyes, go to the optic nerve, they go to the occipital lobe to actually elaborate and process the information and produce a vision. You okay with that?
Yes.
Yes.
Okay, perfect. On another side, I'm going to draw here the cerebellum. The cerebellum are going to be, the function of the cerebellum is the coordination. For example, you are going to walk. When you walk, look at your arms. Your arms are balancing, yes or no? That means your cerebellum is working properly. The cerebellum is balance, coordination, balance, equilibrium. So when you are doing boxing, so these boxers, they are going to try to hit here on the neck, on the back. Why? Because when doing this punch on the back of the head or close to the back of the head, the cerebellum is doing this, and you lose your balance, you lose your equilibrium, and that is going to just be actually part of the sport, right? All right. So that is the cerebellum. Now, this is part of the cerebrum. Okay. Cerebrum. What happened with? Okay, cerebrum. And now we are going to talk about the what? The brainstem. The brainstem are going to be divided in the midbrain, in the pons, and the medulla oblongata. We are going to make emphasis in the medulla oblongata in a few. All right. So from this, basically the brainstem, just to have a very nice panoramic view, are going to be all the vital signs. All the vital signs are going to be located in the brainstem. So brainstem, vital signs, cerebellum, and the cerebrum. So you already know all the actions of all these three components of the cerebrum. We are going to add in a few moments that diencephalon, that are the seeds of the orange. We are going to talk about in a few moments. All right, so let's get started. What I want to finish here is this. Now let's go and buy a pizza. Let's go buy a pizza. All right. So you listen to me, you heard me, right? So what you are using? Your temporal lobe. You're listening to me. Then you're going to look for the list of the pizza. What you like, Hawaiian, pepperoni, whatever you like, right? So you're using your eyes, your vision, your occipital lobe at the same time. You're going to use your occipital lobe at the same time. But you make a decision to make the pizza or your favorite pizza, right? So Sicilian pizza or whatever you like, right? Crusty, thin, very thick, whatever, right? Soft, crunchy, whatever. So that you're using your frontal lobe. Your frontal lobe are actually sharing the information with your vision. What your vision is telling, oh, this is delicious. So that is making a decision on your frontal lobe. So everything is happening simultaneously. Everything is happening simultaneously. We already talked about the occipital, we talk about the temporal, hearing, and the frontal, the decision. Then you're going to decide to walk. You're going to walk to the restaurant or to the area where the pizzeria, pizza place, and then you start to walk. For that, say, oh, where is my wallet? Where is my wallet? My wallet is in my pocket. So parietal lobe start to work. So, but this parietal lobe have a purpose. to find a wallet, to buy what you decide with your frontal lobe, the pizza that you want, and somebody told you that is better, right? So everything is integrated at the same time simultaneously. Then you start to walk. When you walk, you're actually for a purpose. This purpose is to go to buy the pizza. And what you're doing now is to use your cerebellum at the same time. Cerebellum is used when you're standing up already because there's balance and equilibrium. So all this cerebellum has a purpose, the same purpose that your frontal lobe are sharing information with the parietal, with the temporal, and with the occipital lobe. So that is actually what is happening. Whatever connections you find, frontal lobe, occipital lobe, frontal lobe, parietal, frontal temporal, frontal cerebellum, parietal, whatever, all the combinations that you like to think exist in your body, exist in your brain, exist in your brain. Now, we are going to go down the stairs because we are in the school. We are going to cross the street because Signore's Pizza is in front of the street, passing El Camino Real. For that, you need to pass a traffic light. You're using, again, you need to be careful. Also, you're thinking, you need to be careful. Frontal lobe, you are looking the light. You are looking if there is a green light or red light to pass. So that is giving information on the frontal lobe, right? In addition to that, you're going to keep walking. You're doing your equilibrium, your balance, right? You're listening, careful, a car is coming to you. So you start to have, actually, your heart rate start to go up. Your heart rate, your respiration start to go up, and you start to run. All the mechanisms are going to start working simultaneously, including now the brain stem. The brain stem are going to accelerate the respiratory rate, accelerate the cardiovascular. So because of the vital signs. So the brain stem is the center, the brain stem is the center of the cardio, heart, vascular, arteries, and respiratory center. So that is the control. So you need to run, you need more oxygen. So the brain stem is going to do that. So whatever communication you can imagine in any piece, in any centimeter of the cerebellum, and the brain stem exists. We have millions and millions and millions and millions of connections that are going to do the work for us. You okay with that?
Yes.
Okay, so that is the introduction. So let's keep moving. All right, so here we have the diencephalon. So the seeds of the orange are going to be basically the comparison to the diencephalon. The diencephalon are going to compose by three components: epithalamus, thalamus, and hypothalamus. I'm going to remark in the hypothalamus. Remember that hypothalamus is part of the homeostasis. Remember that? The homeostasis are the hypothalamus is the big gossiper. Hypothalamus knows everything what happened in your body, inside and outside your body. So the hypothalamus are going to communicate with the pituitary gland that is endocrine. This is the connection between the nervous system and the endocrine system. That is going to be the homeostasis. That is the hypothalamus. I'm going to put thalamus here just to complete the idea. And thalamus, they have other functions, but hypothalamus is what I want to focus. Hypothalamus are part of the diencephalon. What is the characteristic of this diencephalon? Hypothalamus and thalamus. Those are as well gray matter. They are gray matter. So this gray matter means that are the thinkers, actually the commanders too. They are going to do orders, and that is going to make all. For example, you are lactating. You have a baby. You need to lactate. Who knows that? The hypothalamus. Hypothalamus are going to send orders. I said order because it's gray matter to the pituitary gland, endocrine gland. And what is doing the endocrine gland? Okay, receive the order and said start to produce the prolactin. Prolactin start to produce milk in the breast. So that is another thinker, the diencephalon and the Part of that is going to be the hypothalamus. Hypothalamus. You're stressed out. You're stressed out. Who knows that? Hypothalamus, right? And the hypothalamus is going to send orders to the pituitary gland, and the pituitary gland send the ACTH to the adrenal gland to produce cortisol. You need to ovulate. You're going to ovulate. The ovulation, who knows when you're going to ovulate, is the hypothalamus. Hypothalamus is a thinker. Okay, so I make a decision. We need to produce the ovulation. Okay, pituitary gland, we need to ovulate. Okay, so that receive the order, the pituitary gland, endocrine, pituitary gland, and produce the LH. That is the lutein or lutein hormone that produce ovulation. So that is about what is the cerebrum, the cerebellum, and the brainstem. Be okay with that?
Yes.
Yes.
Okay. So now, you see here the cerebrum are having the cerebral hemispheres, the longitudinal fissure, the lobes, and the diencephalon. That is contained the thalamus and the hypothalamus. The brainstem is going to be the midbrain, the pons, and the medulla oblongata. I'm going to make, when it's the moment, I talk about the medulla oblongata. Cerebellum are going to be basically balance and equilibrium. But brainstem, vital signs. Cerebrum, all what the lobes are doing, right? So we already talked about it. Okay, so to make more complicated, this is the complicated part of the class, is this. That you remember we have the pleura. The pleura is going to cover the lungs. Double layer pleura, double layer membrane, parietal, visceral, fluid in between to decrease the friction. Same for the pericardium around the heart. Similar for the peritoneum that is going to cover the lining of the abdomen cavity. Now, the central nervous system, they have something similar. They are actually called the meninges. What are the meninges? The meninges are 3 layers, not 2. 3 layers, 3 layers, 3 layers. It's like a membrane that you have 3 plastic bags. Open a plastic bag, put a plastic bag inside. Open the plastic bag inside, put a third one plastic bag inside. So you have 3 plastic bags, and then you put all the central nervous system. What is the whole central nervous system? The cerebrum, the cerebellum, the brainstem, and the spinal cord. So all of these are going to be actually surrounding from the tip of the spinal cord up to the cerebrum. 3 layers. In between, as you can guess, in between is going to run a fluid. What is this fluid? This fluid is the cerebral spinal fluid, the CSF, the cerebral spinal fluid. That is very important. The cerebral spinal fluid is there for many functions. It's going to be like a pillow, like a cushion over shock, so over traumas. So that is going to keep the water is the fluid is going to absorb the forces of the pressure. So that is going to protect the brain. Remember, that is surrounding the whole nervous system. Are going to carry nutrients, transport waste product. The main you need to remember is the cushions from shock. Okay? Now, let's start talking about the meninges. The meninges, this is the calvarium or galea, we go call whatever. So if you see here, this is the bone, and here we have the membrane. This is the membranes. These are the meninges. The meninges are going to be covering all the what is that? Redirect notice. What is this? Why? Are you there? Hello?
Yes.
Get over here.
Okay, hello. Okay. All right, so let's talk about the Dura Mater. For that, I'm going to talk about this. Just a moment. It's a little bit off here, but I need to talk about it. Okay, this is a room, okay, whatever room it is, and I want you to imagine that you are in your room, okay, and for this I want you to remember the name of the meninges. The meninges, we have the Dura Mater, that is, write it down, question for exam, is the outer layer. The inner layer is going to be the pia mater. The pia mater is going to be attached to the brain. and the arachnoid is in the middle. All right, so please, I want just you to use your imagination right now. You are in a room. You are in that room wherever you are. Touch the floor with your foot. Touch the floor. Touching the floor, that whatever you have, tiles or you have wood, whatever, that is the pia mater. That is the pia mater. Pia mater. Under that floor or below that floor is going to be the cerebral tissue. Is the brain. Okay? So over the brain, we have the floor that is the pia mater. Now, let's talk about the roof of your room. The roof is going to be called the Dura Mater, is the outer layer. The roof is the Dura Mater. Now, next step, the ceiling, your ceiling. The ceiling is going to be the arachnoid membrane. The arachnoid membrane. Arachnoid membrane is because they have the shape of a web spider. A spider web, what do you call it? Web spider? Web spider. So that is arachnoid.
Spider web.
Spider web. Thank you. A spider web. Web. So that is the arachnoid. And the ceiling is the arachnoid membrane. The arachnoid membrane. Okay, we got it that? The roof, the Dura Mater, the ceiling, the arachnoid membrane, and the floor of where you are there, you are now, is the what? Is the pia mater. Below that is the cerebrum. Now, but there is something weird here. So what is this space between the arachnoid and the pia mater, correct? That space, because it's under the arachnoid membrane, is where it's going to be called the subarachnoid space. Because sub is below arachnoid space. The arachnoid space. This subarachnoid space, this space that you have between the ceiling, the arachnoid, and the pia mater, the floor, this space is called the subarachnoid space. And in that space is going to run a fluid called the cerebral spinal fluid. So it's full of fluid. Full of fluid. You okay with that? You follow me? No?
Yes.
Yes.
Okay. Now, I want to project your imagination that this fluid with these three layers, Dura Mater, arachnoid, the subarachnoid space, and the pia mater, are surrounding the whole nervous system, central nervous system, all the brain, all the cerebellum, all the brain stem, all the spinal cord are going to be surrounded by these same characteristics. Okay, we got it? Okay, so now, for this, I need to talk about the lateral ventricles. So please pay attention to this. These are the lateral ventricles. What are the lateral ventricles? The lateral ventricles are going to be spaces, cavities in your brain. Cavities in your brain. There are going to be cavities in your brain. If you see here, the green is going to be on one side, another side are going to be the lateral the ventricles. I'm not going. If you see here, look at that. Here we have a coronal view of the ventricles. Ventricles are cavities, and these cavities are going to be very important. Why? Because in these cavities we have a structure called the choroid plexus. The choroid plexus. Just remember, we have three layers here, surrounding the cerebrum all the way to the brainstem, etc. Right? And from this, here we have in the ventricular system are going to be the choroid plexus. And that is actually what is going to form the cerebral spinal fluid, the CSF, cerebral spinal fluid. You okay with that?
Yes.
Okay. Now, here we have, I'm going to go here, just a moment, let me see if I don't have another picture here. There you are. Look at this. Here we have in blue, you will see this, the cerebral spinal fluid. And here, as you say here, is the meninges. The meninges are going to be the Dura Mater, the arachnoid here, Dura Mater, arachnoid, and here we have the pia mater. In between, we have the subarachnoid space. Now, here we have the CSF, but the CSF is going to be produced by the ventricular system, through the choroid plexus. Don't forget, question for the exam, the choroid plexus. Choroid plexus. I didn't say choroid membrane. Choroid membrane is in the eye. Choroid plexus is in the brain. Plexus means a group of vessels. Plexus. That's what it means, plexus. Now, look at this. Now, the cerebral spinal fluid is produced in these cavities, the ventricular system. They are going to accumulate. They have different paths. They are going to have a pathway. I'm not going to talk about the lateral, the third ventricle, the aqueduct, the sylvius, and the foramen Monroe, Mangendi. I don't know that. I don't want to know that. So what happened is, look at this. The fluid is going to produce here, go to this pathway in the ventricular system. And then from the ventricular system, look at this. This blue is the subarachnoid space. That fluid is going to drain into the cerebral, into the subarachnoid space. And that is going to be surrounding the fluid all over the cerebrum, the cerebellum, the brain stem, fluid surrounding all the way down to the spinal cord. You okay with that?
Yes.
Okay. So this is what happened is that the CSF is draining from the ventricular system to the subarachnoid space. There is a communication there. That's called the cisterna magna. That is anterior, posterior to the cerebellum. That is another story. The only thing I want you to remember is the concept. The concept is that from the ventricular system that is forming the CSF through the choroid plexus are going to drain into the subarachnoid space. Okay? And let's suppose that one of these circuits or pathway are going to be blocked. Whatever is blocked. So it's blocked. What happened? The choroid plexus start to produce fluid, more fluid, and more fluid, and more fluid, and more fluid, and more fluid. So much fluid, and this is a cluster, right? It's a closed environment. So actually what happened? Pressures are going to increase and push the cerebrum against the bone. And this, especially in newborns, the bones of the skull are not fused yet. They are not fused yet. So any pressure, what they're going to do is to distend the head of the baby. And that is what we are going to see now. There you are. That is what we call the hydrocephaly. Hydrocephaly. The hydrocephaly. The hydrocephaly. Hydrocephaly means water, cephaly, head. So if you see here, this baby is having the head over distended. Why? Because they have a blockage in some pathways. And for exam, you need to know what is hydrocephaly or hydrocephalus. Hydrocephaly is going to be the accumulation of CSF in the brain. You okay with that?
Yes.
All right. So saying that, we are going to actually, all right, so let me see here. All right. All right, so here we have the meninges. This meninges, you see, is like a sheet, perpendicular sheet. That is when the meninges are going to reach the cerebrum and they reach the longitudinal fissure. The longitudinal fissure are going to have like indentation all the way down. And what is doing the meninges are going down and then reflect up. All right. So that's why we have here this structure. Another picture here is going to be, this is here, remove the cerebrum. This is one hemisphere on the one side, and this is the meninges. That is the appearance of the meninges. Okay, more meninges here. We already cut part of the skull. There are going to be this 3-layer membrane, and here covering the brain. So those are the meninges. Okay. All right, so now, let me see here, just a moment, please. Yeah. All right. Okay, so listen. When I told you that there are going to be connection between all the lobes, whatever communication you actually can imagine is going to happen. Okay? Now, if you put your finger on the longitudinal fissure, like here in this picture, you put your finger here, you can literally separate the hemispheres. You're going to put your finger down, 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 but here it's going to stop. You cannot go farther than this, up to here. So what is stopping your finger to go all the way down? And this is one question. The second question, I want you to know, or to tell me, you know that because of this separation, how come the right hemisphere is going to know what is doing the left hemisphere? Because as I told you, everything works together. What is doing the left is going to know the right. What is knowing the right is going to know the left. But there is a fissure there. How is it going to happen? This communication is going to be through this structure here. This structure is like a bridge. It's like a bridge. This bridge is called the corpus callosum. Corpus callosum. Corpus callosum. Corpus callosum. Question for the exam. Corpus callosum, as you know, is going to transmit messages. Obviously, is going to be white matter. White matter. And I'm going to show you the corpus callosum now. Look at that. That is the corpus callosum. Look at this picture first. So you have, it's not like a line. It's like a bridge, literally like a bridge, right? And that is going to interconnect. So you put your finger here, all the way down, stop. What is stop? The corpus callosum. And the corpus callosum is going to have fibers, all the connections that you can imagine that are going to pass from right to left. And that is how the brain is going to be actually communicating or knowing what is happening in the right and the left. This is just a cartoon of the corpus callosum. It's going to go anteriorly to posteriorly. So it's going to go from front anteriorly here down all the way down. So it's like a bridge. Okay, we got it?
Yes.
Okay. All right, so now, sorry, so I'm doing this. I want just to check because the time is very, very short. All right, so now, for this, I want you to remember, this is a coronal view. This is a coronal view. This coronal view, you will see here, that is the left, this is the right, left and right, correct? Now, here in the center, here we have the diencephalon. We have the thalamus and the hypothalamus. The thalamus and the hypothalamus, right? So this is the diencephalon. All right, so just to guide you. Here we have the corpus callosum. Put your finger here. That is the corpus callosum. Here is the corpus callosum. Okay, so now, the fibers that are going to send orders from the cortex, they are going to send fibers, just as an example here, through the white matter. So the white matter is going to transmit to different parts of the brain simultaneously. Very, 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 very interesting. So, and then they are going to pass through the brainstem. Where is the brainstem here? This is the brainstem. This portion is the midbrain. This portion is going to be the pons. And this portion here down is going to be the medulla oblongata. I'm going to focus on the medulla oblongata. The medulla oblongata, and don't forget ever this in your life. So it's the cardiovascular center. It's going to regulate everything that happens with the heart rate, contractility of the heart, the cardiovascular, vascular means vessel, vasodilation, vasoconstriction. Those are the ones who are going to control in the medulla oblongata. And it's the respiratory center. So very important, the heart and the lungs. Respiratory center. The respiratory center is going to control your respiratory rate. Okay? All right. So now, in addition to that, what I want you to remember is this. Look at that. The fibers that are coming from one side of the cerebrum are going to come here all the way down, giving orders, distributing information. And what happened at the level of the medulla oblongata. This is the medulla oblongata. So look at this, what happened. The fibers from the left side are going to cross over on the other side. So from the left, they go to the right. And from the right, they go to the left. At what level? At the level of the medulla oblongata. And that is what we call the crossover. The crossover of the nerves. That is another word that we use is the decussation. 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 Decussation means crossover. So why is important to know that? I will tell you what is important. Do you know somebody who has a stroke? A stroke means that part of the brain is damaged. If the brain damage is on the right side, the effect of the problem will be on the other side of the body, on the left side. If you have a stroke on the left side, you have problems on the right side. Yes or no? You know that, right?
Yes.
Right? And why is that? Why is that? Because the decussation, the crossover that happen of all these fibers, nerve fibers, are going to cross over from the right to the left. From where? From the at the level of what? At the level of the medulla oblongata. You okay with that?
Yep.
Okay, excellent. All right. All right, so talking about my orange, I'm going to delete everything you already know. In this orange, I'm going to divide we are going to have some structures that I want to add here, and this is a review. We are going to do a little bit more decoration soon. All right, so here we have the diencephalon. We have the thalamus, and we have the hypothalamus. That is gray matter. So that is in the center. Those are the seeds. On the side, we are going to have what we call I don't want to do less than the morning, are going to be called, this is called the basal ganglia. The basal ganglia. Basal ganglia are side by side of the diencephalon. These basal ganglia are going to produce the dopamine. Dopamine, what it's doing is to control the movements of your body. So, for example, I'm going to take my soda. Nice soda. Nice soda, right? But if I have low dopamine, look at this. That is what you call Parkinson. And that Parkinson, we will have in pharmacology and med search, you will see that when too much traumas, that destroy the basal ganglia. Yes, just to mention that. But what I want you to know very clear is that on the side of that, side by side, we have another structure that is very important. That is called the what? The limbic system. The limbic system. The limbic system. The limbic system. This limbic system is going to the center, is located in the temporal lobe. It's located in the temporal lobe. In the temporal lobe. And this limbic system is going to be the emotions and behavior. Behavior are going to be the memorization. Limbic system, the memorization. Memory. The long-term memory, the limbic system. Long-term memory, the limbic system. Long-term memory, the limbic system. So the limbic system, as you remember, and that is a review, that the limbic system is going to be divided, limbic system is going to be divided in the hippocampus, what is that? The RAS, the reticular ascending system, RAS, and the amygdala. Amygdala. RAS is actually the reticular ascending system is going to be giving you the alertness. Alert. You're awake. That is going to let you know if you're alive. How do you know you're alive? Tell me right now. Think about yourself. How do you know you're alive? I'm alive. I'm alive. I'm not dead. That is the RAS. That make you awake. In addition, they make you awake. You wake up in the morning, the RAS is working. Hippocampus. Hippocampus is the long-term memory. Long-term memory. Write down that long-term memory. Hippocampus. How you remember that? Hippocampus in Greek means horse. The horses, they have a good memory. So hippocampus is going to have long-term memory. And the amygdala is when you get anger. There is some criminals that they have problems with amygdala, and that actually make change the behavior. And actually, they can be those serial killers, those that killers in series, serial killers are going to be basically some problems on that. There are some findings they found out in the amygdala. So that is the limbic system. All right, so we okay with that? Hello?
Yes.
Yes.
Okay. All right, so what time is it? No way. Can you give me 9 minutes, please? Please?
Yes.
Okay. So here we have the nerve. Please, somebody remind me I need to open the quizzes today for review. So this is something I forgot to tell you. So from 9:30 to 11:30 are going to open the quizzes for review. After that are going to be closed. Okay? All right.
Okay.
So here we have this is the cerebrum, and the cerebrum we have these bulging areas. Can you see these bulging areas? You can see here bulging areas. These bulging areas, and look at this. That belongs to the like the skin of the orange, a very thin gray matter, right? Very thin gray matter. So these bulging areas are going to be called the gyrus. Plural are going to be called the gyri. Sulcus is the line between the gyrus. Each gyrus is the line. That is called the sulcus. In plural, it's called sulci. Gyri, plural, sulci, plural. So now, the question is, look at this. I'm going to show you the cerebrum of a reptile. I'm going to show you a brain of a fish. I'm going to show you the brain of a human. So humans, we have a lot more of these gyri. So why is that? Because, look at this. If you have a line like this, and you have a line like this, you're going to stretch out this line like this. It's up to here. But if you stretch this line, you will have like this. Yes or no? And that is actually doing this up and down in order to save space, in order to have more area for gray matter. Gray matter. We have more gray matter than any other existent animal, mammal, whatever in Earth. So gray matter is actually the characteristic of humans. We have much more gray matter. If we will not have this kind of gyri, right? So if that will be just plain, we will have about 2 yards head size, right? So that's why it's like you with the paper, instead to be like this paper can have a space like this. See how much space is going to occupy this, and how much space will occupy this. We got it? Hello?
Yes.
Okay. All right, so let me see if I finish. I complete this. Okay, so here we have the thalamus. Sorry, the hypothalamus, right? Maintains homeostasis. The limbic system, emotional states, behavior, involve learning and long-term memory. Long-term memory. Long-term memory. Here we have the medulla oblongata. I want you to remark this. Is the respiratory center, the cardiac center, vasomotor center or cardiovascular center, and the contralateral. So that is the crossover. The crossover. The rest are going to teach you another time because it's too much. I need to talk about the cranial nerves, but I'm going to mention just 30 seconds about that. All right, so here we have the cerebellum, and the cerebellum, what I want you to know, You already know what is the function of the cerebellum. What I want you to know that there is actually a structure in between. We have two cerebellums. We have the left cerebellum and the right cerebellum. So these cerebellum are going to be joined together by a structure called the vermis. Vermis. Don't forget, that's question for the exam. Vermis. What is the function? There you are, the vermis. What is the function? Balance, equilibrium, coordination. We have the ventricular system. I want to actually remember those are internal cavities of the brain. What is the internal cavity of the brain doing? Is to produce the CSF, cerebral spinal fluid, through what we call the choroid plexus. Where are the choroid plexus? If this space, your room right now, is the ventricular system, the chandeliers are the choroid plexus. That is where is located the choroid plexus. And that is dropping, like drop, and that produces the CSF, 600 mL. Another time, we will talk more and more detail another time. Okay, so we have the hydrocephaly, the spinal fluid, and the rest are going to be for another time. Just to remember here the cranial nerves, few words about the cranial nerves is this. Cranial nerves. Write down this. Most of the cranial nerves are going to start or begin on the brain stem. In the brain stem. Now, do you notice when you go to the doctor, they give you a neurological exam, and that is something that you're going to do, or you're going to learn, and hopefully I'm going to teach you how to do the neurological examination. The doctor is telling you, can you follow my finger without moving your eye? All right? That is checking cranial nerves. Can you open your mouth and say, ah? So they are checking the cranial nerves. There are going to be different. We have 12 cranial nerves, and each cranial nerve have a different way to assess. So when you are doing that examination, and I'm going to tell you later on, is like, for example, I will tell you, say, open the mouth, say, the uvula is moving. So that's telling you that cranial nerve number 10 is working. Move your eyes like this, without moving your head. Oh, okay, it's working fine. Cranial nerve number 3, 4, and 6 is working, and so on. So, but what I want just to tell you is this. When you have all this assessment of the cranial nerves, it's like literally you are looking inside of the brainstem. So, knowing what is doing each cranial nerve, you are assessing the brainstem. If everything is okay, your eyes are inside the body of that patient, looking that the brainstem is okay. And these cranial nerves, 12 pairs, we are going to learn next class. Where are coming from? These cranial nerves are coming from what? From the brainstem. Thank you. So what do you think about the lecture? Mr. Mark, are you there? Mark, are you there?
I'm here. My camera is just giving me some problems.
Tell me.
I think today's lecture was actually really well. It was actually very interesting and very engaging from the first half when we were learning about the muscular system. And I think today's deliverance was very well. So good job.
Thank you, Mark. Thank you, Mr. Mark. Sherry, go to the jugular directly. Anything that you want to say?
Well, the information was well. I actually understood it. So thank you.
Okay. Miss Arby, please.
Yeah, it was good, Dr. G. I really appreciate your examples. Yeah.
Okay, so review, please. Okay. Thank you, Miss Arby. Elizabeth, please.
It was a lot of information, but the way you explained everything made it very easy to understand. And I actually, like Mark said, I really enjoyed this lecture. So thank you.
Yeah, me too. Sophia, please. I accept critics. Yeah.
No, I thought it was good. I liked the video. It was easier to understand with some of the videos and the pictures you were using.
Very good. Thank you. Catherine, please.
I agree with everyone else. It was well delivered, and I really enjoyed the class today.
Thank you so much. Miss Chelsea, please.
I really liked it too. I like the examples you used to help us memorize the different steps. So thank you.
Okay, thank you, Chelsea. Shafia, please. Miss Shafia.
Agreed with everybody else. It was good, Dr. G.
Okay, thank you so much. Mr. James, please.
I enjoyed the lecture. I like your analysis of everything. And don't forget to open the quizzes at 9:30. Thank you.
Okay, thank you, Mr. James. I forgot already. Thank you so much, James. Steffi, a wrap up, please. What do you say?
I also enjoyed the class, and I appreciate you sharing your mnemonic of the giant cat to help us remember the whole system.
Okay, you will see that this is not in any book, huh? And I will tell you one thing, that I had neuroanatomy for 6 months every day. Crazy. I was just ready to run away. But 6 months is, I have only 2 hours in order to teach you nervous system. 4 hours, because the next is 2 hours more. If you feel that you learn a lot here, wait until next class. Wait until next class. I promise you, I'm going to take you to the next level. Right now, you are different now. From the very beginning, bioscience, at this moment. And that is going to be, before it was like a baby steps, now you are running. We are going to get to the next level every time we are getting into the lecture. And you will learn new things and how to apply that. Right? So thank you so much, everyone. I want to tell you that I really enjoy to be here with you, with all of you. And I want to thank you for all your attention, your dedication, and your effort. Catherine, did you say something? You didn't tell me anything. Catherine, or you already?
I think I did.
Okay.
Yeah, I enjoyed the class. Yeah.
Okay. All right. So I hope you enjoy as much as I enjoy this class. I will see you next class.
Question, Dr. G.
Yes.
For tonight's homework, it says homework 4, chapter 4. It says remotely proctored, and there's a password needed.
Wait. Okay, let me check. Let's do it together, okay?
Yeah. So tonight on.
So thank you for that.
Lecture 4, homework 4, chapters 9 and 10, remotely proctored, and then when I click on it, it says attempt quiz now.
It shouldn't be like that. It should be the previous one. I'm going to correct that right now.
Thank you.
This is the homework you said? This one, right?
Yeah, it says remotely proctored. The homework.
So 12:20 is going to open at 2. Going to be closed on the 19th. The 19th is next Wednesday, yeah? At 11:59.
Yeah.
So now here we have a click here on. Oh no, they already start. Oh no, okay. Somebody start the proctor. So I need to give credit. So I cannot, there's a solution, but somebody already start the quiz. So I can proctor lock it, unlock. Okay. All right. So what I need to do is this. All right. I'm going to grant to that person who did. I'm going to hide it. You will not see it anymore. I'm going to duplicate.
I'm sorry. I think because I tried to click on it and then I saw that it really was proctored and then I got out of it and then that's when I told you.
Okay. Well, let's wait. Let's wait.
I didn't start it, but I think when I hit attempt quiz now, it asked like for me to start the proctoring.
Yeah. So actually, there is no harm on that. That's fine because it shouldn't be proctored. All right. So this is hiding. This is hiding. I'm going to do here. The proctorio is going to actually go away. And then I'm going to edit to be sure that there is no proctorio. Expand. The time is correct. Every question. Proctorio is enabled, so it's not clicked. So now, just a moment, I'm going to save it. And it's going to be unlimited, right? Unlimited time.
Okay, I'm going to check.
Now, just a moment. Now, check it out now.
It's good now.
All right, so I want to know who do the attempts. Otis. Okay. Otis is very always in a hurry. Okay. So so I'm going to give credit to Mr. Otis. That is in the morning. So you have the chapter 1. So I'm going to open quiz number 1. Quiz number 1 is, I need to do for both groups. So I'm going to show, I'm going to start with the group PM. I'm going to start 9:30. Going to do here. What is this? I'm going to need to change everything. 12. It's going to take a moment. 12 January. It's going to be 0. It's going. Yeah. I'm going to be from 9:30. No, 9:30, right? 9:30. 9:30 to 12 to 23:59. Enough?
Yes.
All right, so I'm going to put it in 25 minutes. No. It's nothing. Enable.
So this is just a review. You're giving us access to review our previous quizzes. We're not like retaking it, right?
No.
Got it.
All right, so here we have the attempt. You're going to have immediate after overall feedback. The quiz is open. This is open until tonight. And it's going to be the right answer. I need to put here the right answer. You got it?
Yes.
It's going to be reviewed, right? Okay. Okay, so try quiz number 1. Just a moment, it's loading. Oh, what happened? Oh no, it's 21, 30, 12. Why is not allowing me? No. Cannot be longer updated. Why? Is this proctorio? There is no. Oh, proctorio. No, I don't want proctorio. Oh God, I have a problem here. I need to copy all the exams. Oh, okay, so I'm going to do this. Let me see, is that change? Okay, so let's see. Yeah, I have a problem to review this. All right, so let me see one more thing here. During the attempt, points, right answer. Okay, so I'm going to put it zeros. So you can start now, I guess. Time limits probably is going to be 2 hours. God. Hopefully it's going to make it. No. Oh, there you are. So that was a problem. Can you check your quiz 1 p.m.?
I can see it. It works.
It works? Okay. All right. So I'm going to work on the rest of the quizzes. I need to do 9 quizzes. Okay. All right,
Thank you,
so,
Dr. G.
thank you. Please, during this time, just let me know if you have problems to access to your quizzes. Okay?
Okay, we'll do.
All right. Thank you so much.
Thank you.
See you next
Have a
week.
good night.
G.
Thank
Oh,
you.
wait.
you.
We are going to do tutoring on Sunday, yeah? Even though that we have holiday Monday.
And then
Around
our
the same time, 9 o'clock.
And we are going to do a Tuesday too,
Okay.
9 p.m.
and then Wednesday
Sounds good.
our quiz 4. Is that going to be on Wednesday?
Yes, yes. So for quiz 4, this is a long, I think it's going to be good to do 2 reviews instead of 1.
Great, thank you. Thank you.
Take advantage of,
Good night, Dr. G.
Gracias.
Thank
Thank
you.
you so much. Good night.
Okay, thank you. Good night.
Okay, good.
Bye. Bye, Steffi.